Deepa madam, your voice is not audible.
I guess we are live.
Good morning,
And
madam.
Yeah, madam. Good morning. I hope I'm audible.
Yes, madam.
Thank you.
Good morning, madam.
Yeah, good morning. Nikita madam has also joined. Good morning to her too.
Yeah.
Very good morning,
Good
madam.
morning,
Yeah.
Deepa madam. Good morning,
Good
Nikita
morning.
madam.
Good morning,
Good
everyone.
morning, everyone.
yeah, I guess we should start. Uh,
Yes.
Principal sir is
Uh,
coming, or we should start, and we will address him when he comes.
Okay.
Okay? Yeah. Yes, Saida and Ria, over to you.
Good morning, everyone. We are very glad to announce that we, the team Primers, have kept the promise and came back yet another year with more exciting and enlightening event.
To mark the beginning of the event, let's all stand for the national anthem. You may turn off the camera if you wish to.
I guess there is problem with the audio. It's not coming. Or just mine. Hello. Saida, it's just my audio. Or we cannot hear.
No, we can't hear, sir. We can't.
Okay. Yeah. Fine.
Hello.
A very warm welcome to everyone present here. Also the viewers that have joined us through YouTube. I'm Saida Sayed. And we have Riya Chari. Today, on the occasion of National Science Day, we both take the privilege of hosting our annual event Primers 2022, organized by the Department of Biotechnology, Elphinstone College. It is a pleasure to welcome you all to this wonderful event.
So we have come back yet another year combining knowledge with an element of joy, with exciting talks, research presentations from students and research scholars along with various fun events. So without any further delay, I would like to call Biotech Department Coordinator and the convener of this event, Deepa Hirani ma'am, to formally welcome this gathering.
A very good morning to one and all. I extend a warm welcome to all the dignitaries present over here, guests, faculty of Dr. Homi Bhabha State University, and my dear students, to this Departmental Fest, Primers 2022. Welcome, Honorable Vice Chancellor, Dr. Swati Vaval, Madam.
Madam, you have gone mute.
I'm so sorry. I'll start again. A very good morning to one and all. I extend a warm welcome to the dignitaries, guests, faculty of Dr. Homi Bhabha State University, and my dear students, to this annual Departmental Fest Primers 2022. Welcome, Honorable Vice Chancellor, Dr. Swati Vaval, Madam, respected Principal, Sir, Dr. Vasant Heravi Reddy, Sir, guests, the guest of honor, Dr. Challa, Dr. Amruta Naik, all the teaching faculties of Elphinstone College and Dr. Homi Bhabha State University. I would first like to introduce you to the Department of Biotechnology. Department of Biotechnology was started in the year 2002. It was established and it runs an undergraduate program in biotechnology and a postgraduate program in industrial biotechnology. We as a department conduct a lot of activities, of which Primers is a departmental fest which was started in the year 2009. In fact, Primers is an event which is organized by the students and for the students. It's like a festival where the preparations start a month ago, right from the scheduling to the execution is taken care by the student team under the guidance of the faculty of the department. Now, Primers includes talks from research scientists, research presentations, poster making events, and intercollegiate fun competitive events like memes, reels, quizzes, discussion forums, Petriad, and many more. The main aim of the fest is to give a learning and an interacting experience to our students for their overall development. Here, I would like to add that we have recently started with a Saturday lecture series wherein every Saturday we invite speakers from various fields to give a talk on recent developments in the field of biology, that is, research, innovation, entrepreneurship, etc. This would expose the students to the world outside. So, here we focus in this SLS, we focus on interaction of our alumni with our present students. These alumni have established themselves in various organizations, research institutes, industries, hospitals of repute. So it's like they can share their experiences with our students and enrich them. This would enable our students to make the right career choices and facilitate their progression. Besides that, we have departmental co-curricular activities like seminars, debates, and quizzes on a regular basis. Let me highlight on the research of the department. Our department has a strong research culture. It encourages all our students to take up small research projects right from the year they begin, that is right from the first year. So it's like all the 3 years, first year, second year, and third year, they start doing the research project. Like they start with the literature review and coming up with the ideas in the first year. They do the preparatory work in the second year, followed by the execution in the third year. Now,
Ma'am, your mic is muted.
Ma'am, you
several students, just give me a minute, 1 minute. Principal sir is calling up, 1 minute, please. Saida, please send the link to Principal Sir again.
Sure.
Sorry, I would continue. Because of the research presentation, they go ahead with the research presentation, and they have won awards for their research presentations in several research scholar meets and competitions in colleges across Mumbai. They have been awarded with various awards like students have secured first position and prize at national level research poster presentation in the student outreach program in 2018. This was held by the University of Mumbai Kalira campus. Some students have secured second position in intercollegiate oral PowerPoint research work presentation in Jigyasa competition held at KC College. Our students have also secured third position in intercollegiate oral PowerPoint presentation in Jigyasa 2018 in KC College. One of the students have been awarded Dr. Rajalakshmi Amudan Award as the research best poster competition, which was held at SIS College in 2018. Besides that, in Srujan intercollegiate research presentation competition, which was held at CHM College, one of the students got the best prize. Likewise, to enrich more of the research culture, we have gone ahead and collaborated with Cube Lab under Dr. Homi Bhabha Center for Science Education, TIFR. So Cube Lab, HBCSC, has recognized Elphinstone College as a Cube Regional Resource Center. So again, because of the research activities and their collaborations with other Cube laboratories all over India, our students have developed a strong understanding about the basics of sciences and how do you go about with carrying out research. Cube has awarded some of our students with D.D. Kosambi Young Scientist Award, K.S. Krishnan Student Mentor Award, Cube Kishore Bharti Grant, Obaid Siddiqui Awards for Pursuit for Excellence, to mention a few. In fact, our faculty Mayur Gaikwad was awarded as Q Kishore Bharti STEM Leadership Award 2020 and SK Mahajan Award 2021 for popularization of scientific pursuit. Besides that, students and faculties of the Biotech department are invited as resource persons for workshops and conferences as well. So with this, I would like to add that this year we have introduced MSc in Industrial Biotechnology, a postgraduate program. This is the first postgraduate program in biological sciences in Elphinstone College. The program is unique in the sense wherein the students understand their curriculum in the first 3 years, and in the first 2 years, that is semester 1 to first 3 semesters, semester 1, 2 and 3, and in semester 4 they are exposed to working in the industry. That means we highlight on the industrial training of the students, wherein the students would be given a training in various industries, hospitals, research institutes, etc., so that they have a better understanding of the subject. It exposes them to the hands-on training that they receive over there, and they also have an added advantage of placement. So with this, I would like the event to start now.
Thank you so much, ma'am. Now we would like to call the Vice Chancellor of Dr. Homi Bhabha State University, Dr. Swati Vaval, to give the inaugural message.
Thank you. Good morning to all of you. Dr. Vasant Hedvi, Principal, Elphinstone College and Dean Faculty of Humanities of Dr. Homi Bhabha State University. Srimati Deepa Hirani, Coordinator of the Biotechnology Department and Convener of today's Primers Program. Distinguished speakers, Dr. Amruta Naik, Professor Anil Challa, Dr. Subhajit Sen, all the professors, organizing committee members, and participant students, I'm extremely glad to welcome you and greet you at the inaugural function of Primers 2022, which is being held today, that is 28th February, and on 2nd March 2022. The theme for this year's Primers is Stuck Between Screens, and wherein we are going to focus on teaching and learning science in the online mode during this pandemic. This is the 13th edition of Primers. Primers was initiated with the efforts of Srimati Deepa Hirani Madam way back in the year 2009. And for many of you who may not be aware that I was a part of Elphinstone College that time, rather still I am a part of it. I had worked in Elphinstone College in the microbiology department for 21 years. And in 2009, I still very well remember the first event of Primers that was organized, and it was a great success. And again this year, Srimati Deepa Hirani Madam is coordinating this event, and I heartily congratulate Hirani Madam for the initiative that she had taken and is still continuing, and it is in the benefit of the students that in 13 years this activity is going on, and students in great numbers from Elphinstone College as well as colleges in Mumbai and rather in the country are benefiting from this event. At the outset, I extend my best wishes to all of you on the occasion of National Science Day, which we celebrate every year annually on 28th of February, to commemorate the discovery of the Raman effect by the Nobel laureate physicist C.V. Raman on this day in 1928. Let me brief you about our university. All of you are part of the university. However, there are many participants from colleges outside, for whom I would like to mention briefly about Dr. Homi Bhabha State University. Dr. Homi Bhabha State University is the first cluster university in the state of Maharashtra, which was established in the year 2019. This February, we complete 3 years. The university was started under Rashtriya Uchchatar Shiksha Abhiyan, that is RUSA scheme, of the Ministry of Human Resource Development. Our university consists of 4 constituent colleges, that is Elphinstone College, Sydenham College, Secondary B.Ed Trained College, and Institute of Science, which is the lead college of the university. All these colleges are more than 100 years old and they all have been accredited A by NAC. In all these 4 colleges, we are running several courses. Undergraduate, postgraduate, and doctoral programs are going on in all these colleges. And from the academic year 2021, we have introduced 10 more new courses, and these are masters in various sub-science subjects, as well as we have master in MA in English, we have MA in Marathi journalism. Besides that, we have certain courses which we have started in collaboration with Bombay Stock Exchange, that is BBA and BSc Data Science. Also, in collaboration with Ironwood, we have started a course in sports event management, which obviously we were a little late in doing the procedural part for all these courses. And therefore, unfortunately, we could not get a very good number of students. However, in Elphinstone College, BSc in Industrial Biotechnology, MSc in Industrial Biotechnology and MSc in Information Technology have already started. In accordance with NEP 2020, we also offer choice-based credit system, and we intend to start many more programs in the near future in consonance and in accordance with the new education policy 2020. Deepa Madam has read out the achievements of all the students of the department over the past few years, and I'm extremely glad to know about the achievements, for which I heartily congratulate the principals of the college as well as all the coordinators who have taken this initiative and helping out the students throughout. It is indeed notable to know that the students have won Young Scientist Award and they've also won Student Mentor Awards. And cash prizes and certificates also in national level, also competitions. And I congratulate all the students for their success. It is also notable to know that from the CUBE, the students have participated in various research projects of the CUBE laboratories, and they have been successful in their presentations. As Deepa Madam mentioned, we have started MSc Industrial Biotechnology from this academic year. Though the number of students is less this year, I'm sure that all the TY BSc Biotechnology students would be aspiring to do MSc, and therefore this platform has been made available to them, and I'm sure that next year our batch would run at the fullest capacity. This is the first PG program rather in Elphinstone College, and this program is unique. As Madam said, the first 3 years they would be studying the theory, and in the 4th year they would be given hands-on training, they would be given internship in industries, which will be very useful for them considering their further job prospects. I extend my best wishes for the program, and I now declare that Primers 2022 is inaugurated. Thanking you all.
Thank you, madam.
Yeah.
Thank you, ma'am. Now we would like to call our principal, Professor Vasant Reddy, to give the opening remarks.
Sir, you are muted. Good morning, sir.
Good morning to all. I am very much thankful to Honorable Vice Chancellor of this university for inaugural speech, and it is declared as an open. It's a continuous activity which we are running on every Saturday, and it is very proudful to us that we are linking the students with alumni, and also the very important thing is that biotechnology is a means applied course, which is having not only academic things, but it at graduate level, there is a very good research activities also run, and it is a part of our syllabus. So I am very much thankful, and I'm also thankful to coordinator of this course, Dr. Deepa Hirani Madam, and all the faculty members this course. Thank you. Thanks a lot.
Thank you so much, sir.
Thank you, sir.
Since the beginning, through different theme of primers like History of Sciences, Science Without Boundaries, Science and Society, our department has aimed to bring to light how science plays a crucial role in our daily lives. So let's hear from Srinistha and Vaishnavi about this year's theme.
Thank you, Saida. A very good morning to everyone. The sales of electronics have increased drastically in the last 2 years, and we all know the reason why. The great 21st century pandemic, COVID 19, the era that forced us to rely on our screens more than we could ever have imagined. These are truly unprecedented times, and this pandemic has dramatically changed life across the nation and right here at Elphinstone.
The pandemic brought about a wave of new techniques as well as technology which made us change to a digital way of living. We are all going to remember this change of lifestyle forever. From shaking hands to namaste and from masks being used only by doctors to them becoming a societal norm, this pandemic may have closed our doors but opened many virtual windows. This shift from sudden physical classrooms to online gadgets and paperless assignments has modified our education system for better or worse.
Speaking about societal norms, mental health has existed since forever and will do so in the future. But the pandemic has caused more degradation of mental health than ever before. And studies show throughout 2020 globally there was 28% and 26% rise in major depressive and anxiety disorders respectively. We as students feel that freshers missing out on their college life experiences and others at small moments like having fun with friends, bunking lectures, and having thoughtful discussions with professors, etc., that later make cherished memories, have now been met by the barriers of online communication, thus affecting our health negatively.
And with countries and cities getting into lockdown and lives being pulled to a standstill, the world of scientific research has also taken a huge hit. But now, researchers are moving forward in their own unique ways. Laboratories were shut down, research funding was put on a halt, which in turn made it possible for them to start working from home. The biggest example being home labs. Hence, our theme for this year is Stuck Between Screens. It gives me immense pleasure to welcome you all to Primers 2022, yet another online event to increase your already high screen time. I hope you all can enjoy this enthralling and enlightening webinar. Thank you everyone. Enjoy yourselves. Over to Saida and Ria.
Thank you, Srinistha and Vaishnavi. Surely life in COVID has set a new normal. Though we have learned to talk through screens, it has also given opportunities to explore and learn different skills online. Moving further, I would like to call Namrata ma'am to formally introduce our speaker.
Thank you, Ria. So, it's my honor and privilege to introduce our first speaker, that is Amruta Naik. She is a PhD in virology. So, Amruta had completed her bachelor's degree in industrial microbiology from Rajaram College, Kolhapur. Then her master's degree from in microbiology from Shivaji University. After that, she has been working successfully completed her project under DBT SBI RI in collaboration with IISC Bangalore, where she had worked on a project on evaluation of transgenic cotton containing antisense AV2 gene for resistance to cotton leaf curl disease. After that, she had done her her doctorate research as a doctorate research candidate within the University College Cork, Ireland. where she has done the project on in vitro infectivity of hepatitis C virus in the context of humoral immunity and immune escape. So she had got a fellowship from the university under CTRSP, where she had resulted in 3 peer-reviewed publications, 2 bursaries, and 1 travel award. Now, currently, she is pursuing her post-doctoral fellowship within the field of chronobiology in Children's Hospital of Philadelphia. She is also a good expertise within different techniques during her doctorate degree within molecular biology, immunology, cell culture, bioinformatics, statistical tools. She had many publications during her research also. She is also a mentor, design project, and assist undergraduate student for designing the project itself. So this is what our today's speaker's introduction. So I request Amruta Naik to deliver her lecture. Over to you, Amruta.
Thank you so much, Namrata. Just let me share my screen.
Yeah.
I think someone has to give me a control.
Mayur sir.
Yes, Sachin, can you give your control? Yeah.
Can you see my presentation?
Yes.
Yes, yeah, we can.
Yes, yes, very nice.
Thank you so much. So today I'm going to talk to you regarding working around the clock. You have heard people saying that, oh, my schedule is so crazy. I'm working around the clock. So now when I say the clock, it means in a different context. As we go through the presentation, you will realize it. There are different clocks we all talk about all the time. So I have no disclosure, no conflict of interest to at this moment. So have you heard of biological clock or circadian clock or circadian rhythms? So circadian rhythms are oscillations in various physiological and behavioral processes that occur with a 24-hour period. Wow, this sounds like a really lot of information Monday morning, and that too this early. But let me make it clear to you. When I'm talking about circadian clock, I'm talking about body doing its behavioral biological process at set time, and it occurs every 24 hours, every day at a particular time point. So how does that happen? So the retinal ganglion cells, which perceive the visual information through the light, and as the light enters the eye, it transmits it to brain via their axons. And this happens to a retinohypothalamic tract. The information is given to suprachiasmatic nucleus in hypothalamus. And this information then is given to pineal glands to secrete melatonin, then to adrenal glands to secrete glucocorticoids. And all these secretions, they happen in line with the light dark cycle, which is perceived by eye, and that information which is fed to our brain. Still, I guess it is a bit complicated for you to understand when I say what is a circadian clock. So imagine, so when you have some work planned, what do? You come up with a calendar and put what time you want to do certain things so that you are more productive. Similarly, our body does zillion different things. So it will produce certain juices like enzymes. Then it will produce some brain chemicals. There are genes that have to turn on and off. So that is why our body also has a daily timetable that repeats itself every single day. So the first thing, so that's why the term circadian rhythm. Circadian literally means approximately a day, and rhythm is rhythm. So this timetable is repeated every single day at particular time point. So now you are seeing this body clock. Say with the sunrise, some people get up at sunrise. Then in a healthy individual whose circadian rhythms or whose circadian clock is healthy, all these things happen at these time points. Example, the likely bowel movements will happen at 8:30 a.m. Then as we move into evening, the melatonin will start to secrete, so that it will give a cue to the body that okay, now you are nearing to your sleep time or bedtime. So at this point, bowel movements will be suppressed. So earlier there was this perception that the master circadian clock resides in the suprachiasmatic nucleus. And as our body entrains means it syncs itself from the 12-hour light dark cycles. Naturally, when I say 12 hours light dark cycle, in nature, that means if you go to the woods, there is no influence of electricity. So the sunrise will happen at the time when it happens, and the sunset will happen at the time when it happens. But you won't get interference of the light surrounding us. At that point, in line with that, the brain will get its cue from the sunlight. It will give the signals to different glands in the body, and these signals will be then perceived by peripheral clocks. And then there will different molecular mechanisms will happen at the gene level. But apart from the suprachiasmatic nucleus or master circadian clock, now it has been shown that each and individual organ and cell inside our body has their own clock. Meaning that if you take out this light from a person's life, your liver, lung, kidney, they still will have their biological clock, and their biological processes will happen at a given time point in that 24-hour cycle. So why am I talking about this circadian rhythm or molecular clock? Now you have lived the pandemic, you have survived the pandemic, and you have heard a lot many things about this respiratory infection called COVID-19. Lung, which is an organ that is exposed to these environmental factors, pollution, lung infection, these infectious disease causing organisms. So studying the circadian rhythm or circadian function of lung is of utmost importance. And our lab works on circadian biology of lung inflammation and lung repair and regeneration. So what we do is we work on developing mice models. So in this particular, in our lab, we have studied the circadian rhythm of lung inflammation and lung infection in the context of influenza A virus. So we use PR8H1N1 strain, which is mouse adapted virus. The classic behavior or protection which is offered in circadian way is that the animal will be protected when it gets infected at a particular time point in its life. So one fun fact about mice is that mice are nocturnal animals, meaning that in the night they'll be most active, and in the day they will have their rest period. So what we did was we had these mice and we infected the mice either at 6 p.m. or at 6 a.m. in the morning with this mouse adapted influenza A virus. And as you know, flu is also an acute infection. So that's why we measured their daily weight and clinically scored them. So when I say clinically scored, we look at the mouse and we see if the mouse is breathing properly. Is the breathing laborious? Is the scruffing or the hair on the mouse's skin, are they ruffled, or is it smooth or shiny? So when let me give you some context behind a mouse experiment. I think in one of the slides I have a cage which will show you number of mice we can put. So in one cage we put 5 mice together. So mice, they're much more happier when they are in group. But if they are infected, what they'll do is they will tend to go to the corner and they'll huddle. But if you move one mouse and put it into a fresh cage where there is no other companion for it, then again it will go and hide in the corner. So if you tap on the corner of the cage, the mouse will not move to your stimuli. They in general, mice are very afraid of movements, so they will try to run away up in the opposite direction or in the direction of the sound. So we assign the clinical scores, and in flu or in COVID-19 or any kind of respiratory infection, you lose a lot of weight. So we monitored their body weights as well. So now this is a busy slide and there is a lot more information on slide. So let me navigate you to what's happening here. So if you focus on this particular graph, it's called a survival curve. So on the y-axis, we have percent survival, and on the x-axis, we have day of death. So the animals were infected at day 0. This straight line represents mice who received just control or no virus. And the red line with steps indicate mice who were infected at 6 o'clock in the evening. And the black line with solid dots indicates the mice who received the virus in the morning. So you see the number of steps you see here, that means mice in this group died a lot. That means mice who received the virus in the evening, they have a higher death rate, which was like 70%, 75% higher than the mice who were infected in the morning. And when we look at their weight loss, we can still see that mice in the PM group, or the mice who were infected in the evening, had more weight loss all over from day 1 to day 14 as opposed to the mice infected in the morning. And same with the clinical scores. So from this, we concluded that mortality and morbidity means change in the body weight, from influenza A is under clock control, because we clearly saw that if they got a virus in the evening, they die more and they lose more weight than in the morning at 6 o'clock. So what does that mean? But how is it relatable to lung? So the next question was, first, whenever we are infected, there are two main players. First is going to be whatever natural barrier will be there, that will get activated. Like if you get a skin infection, skin is a natural barrier towards any infection. Then if a pathogen invades, then first responders will be your innate immunity. Then it will taken up by T cells and B cells. So here also, the lung epithelium is constantly exposed to different environmental factors. But before I go into any more details, let me give you a brief idea. At the baseline, that means without any infection, the lung epithelial cells are quiescent, that means silent. They do not divide at all. But once the lung is injured, the epithelial leads charge into lung repair and regeneration. So first we have trachea or bronchi, which has basal cells and club cells. Then we have bronchioles, which lead to alveoli, the actual gas exchanging units. So when you take a breath, this will expand. And when that happens, to equilibrate the surface tension, there are cells called type 2 cells, which will produce surfactant protein, and type 1 cells are the actual gas exchanging cells. So certain cells in the lung epithelium have ability to self-renew or function as facultative progenitor or stem cells. I think stem cell is a terminology which all of you are aware now. So these cells, this is the normal architecture of mouse lung. But after acute injury or lung disease, you can see that you cannot see these air pockets or alveoli here. But as soon as this happens, these cells, they actually come forward, self-renew, and transdifferentiate into different cell types to heal the lung. So now the question was, is clock controlling this healing process? Is the clock controlling the stem cells' ability to heal the lung after the lung disease? To answer that, I again infected the animals either in the evening or in the morning. This is the cage I was talking about. And on the contrary to what you see in movies, no, we don't work on white mice. Most of the mice which we have in our animal facility are C57 black 6 mice, so they have different strains. So anyways, moving on. So we all know that flu is an acute infection, but it could have long-lasting effects on lungs' ability to heal. You have heard now about long COVID, or people who have gotten COVID are experiencing symptoms even after months. So is the clock controlling this lung healing or repair process? And to answer that, I infected the mice at 2 different time points and then recovered them until day 30. And then I did a bunch of analysis on them. So these are the representative lung slices which were stained with a special stain, which were infected either in the morning or in the evening. And then these lungs were scored based on the extent of the lung injury, which is mentioned here. So you can see that the area which is injured in the morning group is much less than the area which is injured in the evening group. You can see more damage in the evening group. So from this experiment, it was confirmed that the clock also controls, the circadian clock also controls the lung repair ability in fighting of the infection. So these circadian rhythms are governed by molecular clock at the genetic level. So what are these genes? Now looking at this slide, you might be thinking, oh my God, there are so many genes. We don't know like which one is functioning, what's the function of each one of these genes. So let's just focus on one particular gene called BMAL1. It's a non-redundant gene. That means it's the only single gene. Other genes like period, it has period 1, period 2, cryptochrome 1, cryptochrome 2. So if you take out period 1, period 2 will substitute for period 1. But if you take out BMAL1, there is no other gene which can function for lack of this particular gene. So the product of this gene are nothing but the transcriptional factors. Those bind to the E-box motifs on the promoters of downstream target genes. So all these terminologies you have studied about or heard about in your lectures. Some genes like BMAL1 act as transcriptional enhancers, and some genes like cryptochrome, they act as transcriptional repressors. So this continuously happens for 24 hours, and it happens via transcriptional translational feedback loop mechanism. This terminology also you might have heard during your lectures. And this particular clock controls how the lung epithelium or airway epithelium reacts after it is exposed to different stimuli like allergen exposure, viral exposures, and how it secretes cytokines and chemokines. So that led to next hypothesis of my project, where I proposed that what will happen if I take out this BMAL1 gene and create a knockout animal as compared to a wild type animal where this gene is completely intact. So to study the effects, I developed 3D cell culture. You might have heard about 3D printer or 3D tissue culture. So these 3D, and we call them as organoids. So these are self-organizing structures which are grown from progenitor cells from the lung, and they are grown in petri plates, and you can see the colonies, like they will develop into colonies like bacterial colonies, and you can see them under the microscope. I'll show you the images in the next few slides. So to first prove the hypothesis, remember I told you that initially it was thought that the master circadian clock resides in suprachiasmatic nucleus, but later it was found that all the organs have their intact clock. So how am I supposed to show that, like how each and every cell has the intact clock? So what I did was I took a mouse whose period 2 gene has a knock-in gene luciferase. So you might have heard about luciferase gene in many of your lectures where you might have heard about bioluminescence. So if I take any organ from this mouse and put luciferin a substrate for the enzyme luciferase, then the luciferase will act on luciferin and it will emit bioluminescence. So what I did here was I collected lungs from this mouse and generated 3D organoids and then added substrate luciferin to see if I will see the bioluminescence. I think I might have to go off the presenter. Okay. So now you can see these individual wells have the lung organoids, and you can see that the light is emitted or bioluminescence is emitted from these individual wells. So these 3D cells or the stem cells which I isolated from lung cells have the ability to function as they have their own clock, and it was proven with this particular bioluminescence experiment where I grew the organoids from these cells, and they have emitted the bioluminescence as soon as I fed them with the substrate. So now when I grew the organoids from these particular mice, so these are the colonies I was talking about. So under the microscope, you can see these colonies which are grown from the individual cells which have ability to function as stem cells. And in the knockout mouse, where it lacks this particular clock gene called BMAL1, you see that there is reduction in colony forming efficiency. So again, this particular experiment proved that the stem cells from lungs, they are under the control of clock for lung regeneration. So in reality, no human being is going to walk off with a knocked out gene, or you cannot knock out a gene like we knock out in mouse. So what are the triggers which will disrupt this circadian rhythm in human? So you might have heard about jet lag, or you might have heard about shift workers, or you might have heard about a lot of antibiotic exposure as a baby. So these things actually disrupt your biological rhythm or circadian clock, which will make you prone to certain infectious diseases as you grow up. So again, these are some cool images which I took from the wild type mouse where the gene was intact, and this is the 3D organoid where the gene was disrupted, and these are the stained organoids from the wild type mouse and knockout mouse. There are a few more images where you can clearly appreciate how these cells have come together and formed a three-dimensional structure which has been taken under a high power microscope. And these are some, again, just some cool images where I have stained the organoids with different antibodies. So the green color is for type 1 cell, and again red color also marks the type 1 cells, and this purple color marks the type 2 cells which have ability to function as stem cell or progenitor cells. So as I was saying that none of the human being is going to walk out as BMAL1 knockout human being. So there are certain factors which will disrupt your circadian rhythm. So again, we did another experiment where we had mouse pups who were less than 12 hours after they were born. They were exposed to 95% of oxygen. So one thing I would like to mention that in real life, we inhale only 10 to 12% of pure oxygen. Rest is mixture of different gases. But suppose a child is born in this world, at that point, with its first breath, the alveoli, they expand. But certain babies, they are not able to do that if their lungs are not completely developed when they are born. So at that point, they are given oxygen externally. But this is very pure oxygen, and the oxygen concentration is 95% oxygen. So, but this oxygen also causes some deleterious effect on their health. So here we wanted to recapitulate the same kind of situation to see if this kind of early exposure is going to disrupt the circadian rhythm. So what we did was we had a control group, where a room air group, where the mouse pups had normal air, and one group where mouse pups who were born they were given 95% oxygen, and then they were infected, then they were recovered until they became adult. In mouse life, adult means 4 weeks after they are born. So then they were exposed to, again, influenza in the morning and in the evening. So again, from the past slide, you are familiar with this kind of setting now, that you can see this is the percentage survival and this is the days post infection. You can see that the animals who were exposed to higher oxygen concentration, they die more, and there is no difference in the animals infected in the evening or in the morning. And then again, you see that this difference is maintained in the animals who are in the room air group or who get normal oxygen. So now you must be wondering, why are we hearing about all these hyperoxia or 95% oxygen exposure and circadian clock? How does it relate to today's lecture series? So I'm getting there eventually. since the pandemic began, you guys were sitting in front of the screen a lot, and you might have heard about blue light. So what does this blue light do? The color of the light, it does affect the circadian rhythms. So how does normal natural light is different, and what aspects of natural lights are perceived by the ganglion cells from in the retina, especially rods and cones? So to give you a context, if you are living in the nature, you don't have access to electrical lighting. Then the day length changes between summer and winter. So in Western world, we have daylight saving, but you must have realized that in the winter the nights are longer, and in the summer the days are longer. So our body clock has to adjust with this daylight changes, and according to the daylight changes, our sleep and wake up time also has to change, and our circadian rhythm has to adjust to this changing daylight. So as a result, our nature has, or we have evolved to have this light sensor, this specialized type of light sensor, and these are called as melanopsin or blue light sensing light sensors, and these are present in our retina. These are not necessarily to seeing the outside world, but this light sensor senses blue light. But why blue light? Because the sunlight is the best source of blue light. When we say blue, it's around 450 to 500 nanometer, in that range. So these light sensors in our retina, they actually are slightly different because they need a good dose of blue light. When I say a good dose, that means, for example, in a full moonlight, your regular light sensors can help you find their way, or you can take a walk. But the level of light is not enough to activate melanopsin. So that means if you stay awake and you are taking a night hike, in a full moon night, it's not going to reset your circadian clock. But now moving fast forward few years, we now understand that the melanopsin blue light sensor actually senses light, bright light during daytime or blue light from the electrical lighting, and then tells our master circadian clock in the brain, depending on what time it is, it might say, hey, it may not be evening, it's actually extension of the day, it's a longer summer day, so please stay awake. And at the same time, it can also send a long signal to this melatonin, which is a sleep hormone, because when melatonin goes up, we tend to feel sleepy. It tells melatonin that, hey, it's not time to go up because it's just a long summer day. And when so that's the way this blue light sensor in our retina connects to our brain to reset our clock, or to tell when melatonin should rise or fall. So now that you have been exposed to a lot of blue light during COVID, since your lectures were all virtual, but we also had to consider the fact that since you had stay at home orders or everything was restricted, your source of entertainment was television or laptop or your mobiles, and more you got exposed to blue light, more you slept later in the night. So there was a group called by Stone et al., which studied the effects of online school versus in-person school. So they enrolled students who had in-personal learning, and who had, after the state of emergency, who underwent the remote learning. And they looked at their actigraphy. So for actigraphy, what they did was they gave a watch which was worn on their inactive or the hand which is not that active. So some people who use their right hand, they put their watches on the left hand, and some people do are left-handed, they will put the watch, this actigraphy watch, on the right hand. Then they used dim light melatonin onset in the children who attended in person versus remote learning. Again, there are lots of box plots here. On the y-axis we have sleep duration, and on the x-axis we have different groups, where in the school days where they have in-person school or remote, or in the free days where there are no lectures like Saturday or Sunday, people who attended in person versus remote learning. And on y-axis we see the sleep duration. So students who have in-person school, you can see that the sleep duration is approximately between 8.5 hours to 9.5 hours. But who are attending the remote classes, it has significantly increased. Then again, when you look at the sleep onset time, people who are attending school remotely, they're falling asleep pretty later, like past 10 o'clock, towards the midnight. And their wake up time also is then again past 8 o'clock in the morning, rather than 6 o'clock, which we see here for the people who attended schools in person. And even in the free days also, there is delayed onset of wake time and delayed onset of sleep time. So what does it say about, in general, that there is a relationship between circadian phase and sleep wake time during school days? So here we see the open triangles, they represent remote learning, and these closed circles, they represent in-person school. So we see that clearly people attending the remote school, they are sleeping later in the night, even during the school days and even during the free days. And again, they are getting up later because they're falling asleep also pretty late. And it hasn't changed either in the school days or in the free days. So what's the conclusion of that experiment? So current built environment promotes a delayed circadian rhythm, and this often conflicts with alignment of our social clock. And what they observed was, on an average, students who had this remote learning, they approximately went to bed 30 minutes later than those who took the course pre-pandemic. And there was changes in the natural sleep cycle. So I'm pretty sure now you understand why we used to hear that लवकर निजे लवकर उठे त्याला आरोग्य धनसंपदा लाभे, because all these things are happening in sync with the nature and with the circadian rhythm. So I'm not sure up to what extent still the schools or colleges in India are functioning remotely. But is there a way to correct your circadian rhythm or to be in sync with the nature? Yes, there is. You can limit your exposure to bright light or blue light 2 hours before you go to bed. And this has to happen every day. Again, second thing is, you can entrain your body to external environmental cues by time restricted feeding. You might have heard about this fancy terminology called intermittent fasting. So meaning that if you eat your breakfast, lunch and dinner at a specific time only, your body will get reset to a particular biological clock or circadian clock, and you will lead that will help in having healthy changes in your lifestyle. And third thing is exercising. You can also exercise to keep your body healthy, but if you exercise on a particular time, it helps alleviate the symptoms of jet lag, and it's been proven as well. So I hope I have opened your mind, eyes, about effects of circadian rhythms on your health, and you will follow it up to some extent in your day-to-day life. And that's it for today. I hope you enjoyed the lecture and I was able to convey most of it. And thank you so much thank you so much, organizers, for allowing me to share my research. And hi, Nikita ma'am, it was really good to see you here. It's really nice to meet people who I have met when I was back in India. I can take questions if anyone
Yeah,
wants
yeah,
to.
there are some questions. So the talk
Yeah,
was really very nice. Means your postdoctoral work is really nice regarding the virus you have. Means on the virus, on the animals, the experiments you have done. It's really means encouraging even to our students also, because some of the students they are working on snails means in our lab also. So for them also it will be very helpful. So
thank you.
there are most of the questions there. So one
Yeah.
of the student asking, just a minute. So what is the role of a light in a circadian rhythm?
So we were developed, like, before all these electronic equipments or electricity came, we used to perceive the external environmental signal, and our body used to synchronize our all biological habits as per when the sun rises and when the sun sets. But since we have developed even more, the external light sources have messed up our circadian rhythm. So if you ask what will happen to the blind person, that blind person's circadian rhythm will be totally different than a person who can perceive light through their retina. Their circadian rhythm is going to be different. Even if so, for example, in my study, when we want to decipher the role of circadian clock at the organ level, we don't want the light to entrain the organism or to sync the organism with external cues. So what we do is we keep these mice in continuous darkness. So what rhythms or what signals it will get is from their own internal system rather than light.
So another question is, does the circadian rhythms change with respect to age?
They do. This is a really good question. So as you age, the circadian rhythm changes, and actually it might make you more prone to certain infections. And there had been studies that many people who get it's not compulsory in India to get flu vaccine, but here in the US it's compulsory to get flu vaccine every season, every year. But older people, they produce less antibodies against influenza as opposed to younger people. So there we have done research that you could actually entrain person's ability to produce antibodies by immunizing them at different times of day, and you can actually increase their ability to produce antibodies, especially in the older population. But this is a really good question. You can't do much since you're aging, your all the biological system is changing, but definitely it affects your circadian rhythms as well.
So, sir, this Arunan sir, he is asking one question. So, any studies on get watchmen during night schedule? So, what is the effect?
So, not on the watchmen, but there had been studies on the shift workers who work in the night. I don't know in Indian people, but they have done these kind of studies in shift workers or jet lag, like people who frequently travel between the countries. They always they have phase shifts. So the time at which they will go to sleep will be totally different than people who work early in the morning versus people who work in the evening. Their circadian rhythms are pretty much. Why go far away to like gate watchman? Think about IT people who have like absolutely messed up circadian rhythms because they are awake at 8 o'clock in the morning. They go to the sleep, go back to sleep at 1 o'clock. Then again they are waking up at 6 o'clock. My husband is an IT worker, so I know his circadian system is totally messed up.
Okay. Mayur sir, you want to ask? Deepa ma'am.
Yes. So Amruta ma'am, in Cube, we are doing this experiment on fruit flies. So we are trapping these fruit flies at different times of interval, and some students have got that there is a peak of these fruit flies when they come to the trap. So in the morning and in the evening. So around morning 11 to 1 and in the evening 5 to 7. And this experiment was done by school students, and this peak changes as the season changes. For example, if you talk about March, which is solstice, and when we talk about March, which is equinox, and when June, which is solstice, so the peak changes from solstice to solstice, from March, June to December solstice. So the second thought, what we are going to do is do this across the India, from Kashmir to Kanyakumari, from eastern region to western regions, on different latitude and longitude. Is a different latitude, and Kanyakumari is a different latitude. It's higher latitude. So do you think the activity pattern will change according to the latitude and longitude?
There is a possibility because of the change in sunrise and sunset. You might be able to see the differences in the peak based on the season, at what time you are doing your experiments. It's gonna change a lot. You can definitely look at Amita Sehgal's lab at UPenn, because they work on fruit flies, and there is one more lab called Alim Velji. So these people work on fruit flies to study the effect of circadian clock. So definitely this is the coolest experiment, and I'm glad like people know about all these things. I came to know about the circadian system when I started my postdoc, so I was pretty late to the party, but you guys are much ahead in your time now.
There is a follow-up question, if you don't mind asking.
Yeah, sure, go ahead.
The thing is, when we talk
Deepa
about
ma'am.
this genes related with circadian rhythm, let's say there was studies on eclosion pattern when they discovered about clock gene and per gene. Do you think those the same genes play a role in different circadian rhythm behaviors, or there are different genes for different behavior as such?
So do you mean that the physiological behaviors you are seeing, are they related to a particular molecular signature in general, or it differs from organism to organism?
Yeah, so do you have if you talk about, do you have any generic genes which are related with the circadian rhythms, or which controls mostly all the circadian rhythms? If you talk
So
about
BMAL1 is the gene which is the non-redundant gene. So if you take out that gene, the organism becomes arrhythmic. So that means it no longer can perceive the outside signals, and it will not have 24-hour period. So so in case of mice, we measure their activity, since they're most active in the night. So you can see when they are active through our infrared rays. So they'll pass through the maze a lot of time in the night when they're active, and you can see the disturbances, and they are plotted in a different way. So you see a clear-cut 12 hours activity and 12 hours rest period. But when you take BMAL1 out, so it's very messy at that point. So that's how you know that a particular gene is responsible for that. But if you take out PER1, then PER2 will substitute for its function. So that's BMAL. But again, if you take out BMAL1, it also has non-circadian effect. That means these kind of genes also have been involved in development of animal. Like if you take out the BMAL1 earlier, or before the mouse is born, then it could result into early aging. So the mouse ages much earlier than its life period, or it could maldevelop the organs. So those are also the effects, like non-circadian effects, which are governed by this BMAL one.
Yes, I guess Deepa madam was to ask something. Deepa madam.
That was a very interesting talk, madam. Um, you explained about the effect of food, exercise, and sleep on stabilizing the circadian clock or the biological rhythm. I would like to know, as you were mentioning about intermittent fasting, that is the time at which you eat the food is very important to stabilize your biological clock. So, is it just the time, or it's even the type of food that you eat? Do superfoods or other foods have an effect on our circadian rhythm?
That is again a good question, because we also do experiments by feeding mice high fat diet. So when I mean high fat, means we don't feed them cheese or sausage, anything like that, but they have a special food where the fat content is a lot, and we do see that it messes up, like they have more body weight, and it differs from males to females. So even if you have a time restricted feeding, you eat at particular day, it has to be a balanced diet. You can't just eat chocolates and ice cream within that particular time and say, okay, my circadian rhythm should be back to function. Everything has to be in moderate amount, and it has to be a balanced diet. So definitely that will help in long term.
Thank you.
Yes, do we have some more questions, Namrata madam? I guess. Yeah.
Yes, actually, we have more questions. So, as the another speaker is there waiting. So, just one last question I want to ask. I will mail you the remaining questions. That will be
Yeah,
okay,
sure.
Amruta?
Yeah, that's definitely okay. You can get
Uh,
in
so
touch with
then
me
he is
anytime.
asking that when we take a vaccine, so we are taking a either in the morning as well as in the evening. So do that play a role? The circadian rhythm will play a role within vaccination.
It definitely, because I also had done an experiment where I had vaccinated mice in the morning and in the evening, and I do see a difference in their antibody titer. And recently, there was a paper published with respect to SARS-CoV-2 vaccine in Chinese health workers, because it was easier to track at what time of the day they got vaccinated. And people who got their vaccines early in the morning, between 8 to 11 to 12 o'clock, they had much more higher antibody response and the antibody producing cells in their system than people who got later in the afternoon. Having said that, it doesn't mean that if you got your vaccine in the evening, you're not immune to the virus. You are still immune, but people will have better antibody titers if they get their vaccine early in the morning. That is your peak activity period, before
In
12
the
o'clock.
morning, it
And
will
it,
be
it
more
will
active.
differ from virus to virus, though. You cannot generalize it, like this has happened to SARS-CoV-2, or this has happened for the influenza vaccine. There are opposite effects in terms of hepatitis C virus. It has been studied. So it totally depends, varies from virus to virus. So you have to keep that also in mind. But definitely circadian rhythms play an important role in vaccination as well. And it's been studied for at least flu and for SARS-CoV-2. If you get it in the morning, it's better for you. But if you have had your vaccine in the evening, it's not end of the world. You are still immune to the virus.
Okay. So one thing means now we are working on circadian rhythm. So do they go on yearly and yearly the same things? Means
So
suppose one situation it is.
yeah, that is also a
Please,
really
Amruta.
good question. So if in an ideal world we are never exposed to pollution, we are never exposed to blue light, then our body is supposed to function based on the how Earth rotates, and based on when sun rises and when sun sets, each and every activity. The third slide, which I showed, that at 6:45
Six.
some things will go up, at 9 o'clock your melatonin will rise. So there is a group in California, from Sachin Panda, and he has studied the circadian rhythms in I think Aboriginal people, where there is no light. So all these people have natural food sources. They are dependent on sunrise, sunset, which happens naturally. They have the perfect circadian rhythms. All the people who live in the city, they don't have perfect circadian rhythms, unfortunately, because there is blue light and there are other effects which are happening. I don't think so anyone goes to bed at 10 o'clock or 10:30 anymore.
Okay. Okay, nice. Good. So we'll have means most of the questions they are. So I will share you on the mail.
Sure.
Saida, you can take over then. Saida?
Thank you so
Yes,
much, guys. I really enjoyed the question answers as well. I think I was able to convey you the importance of circadian rhythms. And best luck with everything.
Yeah, thank you,
Thank you,
ma'am.
ma'am.
Thank you.
So I will now hand over to Dalraj to give the vote of thanks.
So thank you, Amrita ma'am, for giving such an informative and thought-provoking speech. We truly appreciate your insightful words on the topic of circadian clock, which mentions our body's timetable that repeats itself every day. Personally, I liked when you talked about the earlier perception of master circadian clock, which has now been debunked. I would also like to bring up your interesting experiment on the mice and their mortality and morbidity from influenza, as well as the relation between blue light from screens and circadian rhythm, especially during this pandemic. So thank you again, ma'am. I, Dhanraj Sribon, on the behalf of Biotechnology Department, would like to extend my gratitude for sparing your invaluable time for us and gracing this event. I can strongly state that we all are going to take your words with us long after this event. Thank you.
Thank you, Dhanraj. Now we would like to call Mayur sir to formally introduce our guest of honor.
Yes. So, welcome everyone. Thanks, Saida. So today we have a very distinguished personality with us, Dr. Anil Kumar Challa. Currently, he is a senior scientist in Department of Life Sciences of Natural Sciences at Srinagar University, Delhi. He is adjunct professor in Department of Life Sciences at University of Alabama. Dr. Challa obtained his training in molecular genetics and developmental biology and is currently involved with a number of research projects focusing on generation of genetically engineered model organism like rat, mouse, zebrafish, and etc. to understand human disorder. He provides technical and instructional support in this project. He is also involved with the grant writing and professional development activities focused on undergraduate and high school biology education. He did his bachelor's in agriculture in agriculture from Banaras Hindu University. He completed his PhD from Ohio State University in molecular genetics in the year 2003. His doctoral research involved the identification and characterization of receptor protein involved in embryonic development, especially exon guidance during neuronal development using zebrafish as the model system. Then he did his research as postdoctoral fellow at many laboratories like University of California, San Diego, Medical College of Wisconsin, the Ohio State University. He was also visiting scientist in the University of Texas Health Science Center. He co-founded a startup biotechnology company in Hyderabad, India, where he set up the laboratory and directed the education and outreach activities. Then he did short stint as a senior research scientist at University of Life Sciences. Now it's called Dr. Reddy Institute of Life Sciences in the University of Hyderabad campus, where he established the first zebrafish facility to support research in drug discovery and testing. His work is published by many reputed research journals like Science, PubMed, Current Science, etc. His research interests are developmental genetics, genome editing, and undergraduate biology education, That's how we know each other. From my first year and second year undergraduate, I used to write Anil sir lots of mails because we were in the Cube mailing system. So, and he used to take time and used to answer like such a long answers, not giving direct answers, but again asking for more inquiry. And he used to give such a precious time for an undergraduate, first year, second year undergraduate student. So that's incredible. So without further ado, I should call Dr. Anil. Anil sir, are you there? Yeah.
Hi, Mayur. Yeah, I'm here.
Please, yeah, so please take over. Yeah.
Thank you very much for. This has definitely been a very pleasant surprise hearing from you again after many years, and it is truly a delight to actually be here and in the midst of all of you. I wasn't quite sure, so I kind of tried to blend in some of my thoughts with the theme of today's your festival, the Primers 2020. So let me share my screen. Let's see if I'll get it right. Can you all see my screen?
Yes, sir, we can. Yeah, we can very well see your screen. Yeah.
Quite, let me see.
So while presenting, you can be on the this thing.
So I think there is some issue, right? So it's actually mirroring the whole thing. So okay, so I think this should be better. So I have a couple of videos which are kind of important, and hopefully they will come through. So I'm going to reflect on an evolving experiment in undergrad biology education. As Mayur mentioned, I've had a very strong interest in undergrad education for quite some time. And I'll tell you about a recent experiment which started off in 2019. But before I do that, I wanted to give you an outline. So I'll tell you about the motivation behind my current experiment, how the current experiment started and how it transformed during the pandemic, and leave you with some final thoughts. So to begin with, I was really motivated by a good study, a reasonably well done study called Vision and Change in the US, funded by the National Science Foundation and the American Association for Advancement of Science, the same organization that publishes the international magazine Science. So this actually came out, this document, Vision and Change, came out as a consequence of a 2-year exercise where they included a lot of people, the stakeholders, the teachers, university administrations, policymakers, and people in the industry and basically the employment sector. And they asked, what should be done to improve undergraduate biology education? And they came up with a call to action. This particular document went through several revisions, and there have been several successes. While I was reading Vision and Change, I was getting to know about their accomplishments or their efforts. I was at the University of Alabama at Birmingham, and there was an opportunity for change. So there's a very, very strong undergraduate science and technology honors program at UAV. And the first year, students actually go through a two semester course series. So the first one, so that is the first semester in first year, is Science and Technology 199, Introduction to Scientific Process. So in this particular semester, students they actually discuss basic concepts of scientific methodology with a goal of analyzing a scientific publication. And then it goes on to the second semester where they are introduced to research approaches. So I will not talk a lot about the research approaches, but stick to the first part. So when I first entered a classroom for STH 199, and I saw students struggle, remember this is high school students coming into college for the first time, and the first semester they get a research article, and they were supposed to, for the most part, it was a relatively recent paper. you can imagine the struggle that they would have faced, right? So I did not think it was appropriate to give a cutting edge research article to first year students. But to my utter surprise and delight, towards the end of the semester, in an exposition, students presented their understanding so well that I was blown away. And fortunately, for the next several years, I've been part of this program, and I saw year after year how well even first year students get introduced and master to some extent how to understand a research article. So there are several methodologies, but one of them is called the CREATE method, which was started by Sally Hoskins at the City University of New York. And they claim the idea behind this is to transform understanding of science through primary literature. So this is the first research article that they publish. As you see, Ross name, Leslie Stevens and Sally Hoskins published this on selective use of the primary literature transforms a classroom into a virtual laboratory. So I felt that having seen the success of this particular method, I thought, can we replicate this in India? Now, I talked about this with a few people because I was not able to directly access students. And a resounding message or a feeling that I got back from colleges and many teachers in Hyderabad, because that's where I was based on, so this is not possible. It is not likely that they will do it well. They need a lot more basic understanding before they even think about tackling current research articles. But somehow, having seen the success in the US, I felt, and having seen that the student aptitudes and personalities were not very different from the students I met in India, I felt this could potentially be done. So we started off in 2019. I conceptualized a 6-week program, and I specifically wanted only first year life science undergraduate students. And I was able to meet with students at St. Anne's Degree College in Hyderabad, and I basically made them work in teams. Now, as you can imagine, there were a lot of suggestions that they said, why don't you start at the master's level? Why don't you include the first year PhD students? But I really wanted to, the philosophy behind this was, if first year students actually are able to access their own capabilities in terms of understanding cutting edge science, they are very well placed not only to understand, to get deeper into learning new areas of knowledge, but also breeze through their course curriculum. So, and as I mentioned, all students were given a recent research article, and we used elements of the CREATE method, not the entire process, which is very elaborate, but we used multiple elements of the CREATE method to introduce and understand the research article. So eventually, the students prepared and presented it to an authentic audience. And the authentic audience I want to emphasize here is, these are researchers at a research institute. So it is not just in a college environment where they are presenting it to peers and teachers, but they now had to face a new audience, which is involved with research on a daily basis. So this was the paper, Tumor Microbiome Diversity and Composition Influence Pancreatic Cancer Outcomes. So the program started in September 2000, I think August 2019, and this paper was published in the August issue of the journal Cell. So I thought you should listen to one, towards the end of the presentation, there was some discussion going on. So here it is. So Mayur and whoever is doing the technical stuff, can you please tell me if the audio is working? Give me 1 second, I'll play it.
Yes, it's working. It's working.
Is it working?
Yes, sir. It's working.
Yeah. So this is about a 2-minute clip. So I want all of you to watch it, and we can. This is so that you can reflect upon the outcomes. Remember, this is the end of the 6-week period. In fact, they've only been reading this paper for about 4 weeks. It was quite intense. I would go to the college 2 to 3 times a week, and I'm more than happy to share the protocol that we followed in terms of how to engage students with this particular paper.
So if somebody has an HIV infection, because our immunity is suppressed by the virus. That's an indirect indicator that there is virus. So we can, each of us can give blood and they can do a test. They count essentially, they count the number of CD4. That's most likely what it is.
So, sir, basically from this study, we conclude that our main aim was, the study's main aim was how LTS are surviving modern disease, right? And we had to find what is present in LTS that is enabling them to survive. So basically we have found...
I guess now we can't see the video.
Sorry?
I guess we can't see the video now.
Sorry, got sorry about that.
Yeah, we can hear it, but we can't see, I guess, now. Earlier we could have saw that.
Is it better now? Can you hear it?
Yes,
And
I can hear, we can see. Yeah.
which enables them to favorable treatment, and also they have high CD8 plus T cells, which enables them to react to immunotherapies and chemotherapies because they act as immunoactivation cells. So
We just thought that this can be the measure. Still, there is research is going on.
So do they talk about what bacteria, what are the species?
Yeah. Sir, about figures of concentrations 1 and 2, they have talked about it on in figure 2. I just know one of it, and it's streptomycin.
So the idea is not to, so hopefully you got a glimpse of it. You didn't have to understand every bit of what the students were saying, but this was a case with multiple, there were multiple teams presenting some of the stuff. So we tried to do it in an economical and yet an effective way. Sorry. So going to the next one. So, but then very soon after that, you can imagine, that was September 2019, but by March 2020, we already, we were, the pandemic hit and we were in lockdown. So some of these students came back and said, sir, why can't we do this? I mean, now that, we can't really go to college, can we continue doing this stuff? So we adapted the September 2019 experiment or the model to the online platform. We started initially with students in Hyderabad, but organically it spread by word of mouth and included students from Nagpur, Varanasi and Chhattisgarh and a couple of other cities that I failed to recall. So again, I will indulge you in watching. This is slightly a little more elaborate, but it will be useful hopefully for us to discuss towards the end. And I intend to keep this presentation short so that we have enough time to talk about the pros and the cons and your any concerns or any thoughts from your side. So here you go. I hope you can see the video and the audio as well. This is after about 6 months of doing this. It was by chance, it was a random picking from my collection of videos. It's an online presentation of students. This particular group is presenting a second paper in the series. So there were multiple teams presenting every week. So we were meeting every week. And this particular team is presenting their second paper.
We are team PCR, that is Rrna, Chaitanya and Riya, and we welcome you. So today the research paper we are going to share is CRISPR-mediated activation of a promoter or enhancer rescues obesity caused by haploinsufficiency. So let's start. So first we will know what is haploinsufficiency, because it is the only problem which is addressed in this research paper. So as we all know, there are 2 alleles or 2 genetic copies. So they use the neuroblastoma cells of mouse hypothalamus, which are known as neuro2A cell line, which is known as, and they will just transfect this cell line with this Cas9 system, that is CRISPR system, and they will see how much upregulation is occurring in vitro. So after the cell line has grown for 48 hours with this CRISPR system, its mRNA expression was measured by quantitative PCR. Let's see. Now this is the graph about SIM1 expression, and there are 3 types of individuals. One, it is control, it has no guide RNA. This one has guide RNA which is targeting promoter, and this one has targeting enhancer. So this promoter targeted system upregulated CIM1 expression 13 times the normal one, means with respect to the control, and means those cells which were targeted by enhancer guide RNA CRISPR system, in that the CIM1 upregulation was 4 times that of this control system.
And so CRISPR system can rescue a haploinsufficient phenotype in vivo. These results provide a framework to further develop CRISPR as a potential tool to treat gene dosage sensitive diseases. Thank you. So here we have come to an end of our presentation. Now you may ask any questions that you have. Once again, thank you so much.
What was so significant about an 11th week?
The difference in the weights will be observed after 11 weeks. So before that, maybe there was not any significant difference in the weights. Yes, sir. So actually, in this paper, there were many new things I learned about, like knock-in mice, the microdeletions, and homolog HDR. Actually, and I enjoyed the entire process. Actually, about the discussion, about making the presentations, the slides.
So the part which I learned from this paper was one cannot understand one research paper by only reading that paper. It is compulsory, or it is, I can say it is natural, that we have to prefer other things, other papers related to it, to collect small things about this paper, and that also answers the...
I agree.
Yes, that also answers the third question, that if we were having more time, then we would have collected more other articles, other research papers, because we were referring to other articles also, because we were not understanding many things, such as how this MJR is expressed, or how these vectors are used in this RAAV. So we were just jumping from here to there, searching other articles. And that was also fun part, because it was very challenging paper. It was confusing us also. But it was good to work on this paper and all other related studies. It accelerated knowledge very much.
So I would say that this time I was a little less nervous before the presentation. And some of the suggestions that came to us after our last presentation, we have worked upon that. I would like to tell you an example, take this slide which is being presented. The process is given on the left and the diagram is on the right. So this time we chose to use the animations wisely. First we explained the process, and then the animation came and the figure came. Last time what the mistake we had done was we introduced both the things at the same time. So people were confused, like we should pay attention to the thing on the left or on the right. So some such small improvements we have worked upon this time.
So this is a very badly put video. It was a 2-hour session that I condensed into 5 minutes to just give you an idea about how one of these meetings was went about.
We are team T.
So let me go to the next one. So the experiment is still in progress. In fact, I think we'll have enough time to discuss. I thought it will take a little longer for me to bring you till this point. So we have included a few other features in the Samyojana model, where we have a book festival at the end of the semester when everybody gets a chance to present their research paper. Each student gets biographies or autobiographies of scientists or discoveries or inventions. And we've had 2 of those sessions, and it was a fantastic experience. And from the student feedback, they were really excited. this actually brings in the idea that science is not, it's a very social process where it's not just about information, knowledge, experiments, and results, but how people are involved and people's lives are involved. We did a small tweak in this. I'm still working on this to create groups where experienced peer mentors will help new participants experience Samyojana. We called it the Taruna Samyojana, and there was some very interesting. There was very good feedback from both the new students and the experienced students, because they got a chance to act as mentors to the younger students. So we are still working on it, and now as we speak, it is entering its third year. So we will have to wait and see what new learnings we have. So I thought I will spend the next few minutes in talking about some tools that we used and some thoughts on how to enhance learning. One thing I want to mention here is, and I'll probably mention it again a little later on one of the slides, is the greatest feedback I got just from the experience, not in terms of words, is how quickly students were able to pick up something. And it's probably not a surprise. I think all of us, I am talking to people who are already experienced with this stuff, both students and teachers, that if the right kind of motivation is set, the right kind of resources are provided, and a decent amount of support is there, I think students are going to run for it, and they will actually learn things much more efficiently than what I could have done through standard traditional practices. So it was very delightful to see how students are self-motivated to learn and how they're able to help each other to get a greater understanding. So learning is also a social process, and so we started using, some of you might be aware, this is a tool that was developed at Harvard, and one of the main people who's involved in this is Dr. Eric Mazur, who probably some of you are aware of, who's famous for his very well viewed YouTube video called Confessions of a Reluctant Lecturer, or something of that on those lines. How he discovered that lecture was very ineffective, and how peer learning is extraordinarily valuable and effective. So perusal aims to change the nature of reading. Now I use perusal basically by putting the papers, and I'm more than happy to share you some screenshots of how student discussions happen when I upload a research article, right? And how students are reading the paper and discussing. Where this tool, it actually enables students to go from traditional solitary experience of reading to an engaging and collective one. Perusal aims to change education, so all students do the reading, come to class prepared, and are motivated to do so because they care about the content. Typically what happens, and I think this is something I have noticed, is when I come to class and I'm trying to teach something for the first time, there's no prior preparation. So I'm sure some of you have, I think it's already happening. I haven't taught any traditional courses during the pandemic, but I think it's already happening in terms of a flipped classroom, right? So you give the material to students ahead of time that they engage with, and then a greater discussion happens during the class time or the meeting time. So I think that is one big learning for me. And I also actually get a chance to see how much students are able to handle the material, which is on perusal. And if they have any prior challenges, I can address specifically those things. And I don't have to assume that they're understanding or not understand this given material. So with all this, with my experiences, and I think where we are going ahead, I think many of the students will probably vouch for this. During the pandemic, I've had more students do Coursera courses, edX courses, all kinds of online courses by themselves, because at least some of the students I was dealing with, they didn't feel challenged enough with normal college curricular material. So they went out and did this. They looked for courses and they finished them successfully. But they still needed help, because it was not always that they were able to comprehend everything by themselves. So why Samyojana? Why bring in research articles? Why challenge them with something extraordinarily complex? And one of the things I think we are all realizing and where we are heading to is from remembering information to creating knowledge, right? So because now literally everybody has, or at least most of us who are able to access the internet and who have a smartphone in our hands, literally have information at our fingertips. It was a phrase probably, I don't know when it was coined, but today that particular thing literally has become a reality. And many of you probably know about Bloom's taxonomy. So I think it's timely. It's been a reasonably optimal time for the past maybe 10-15 years. But the pandemic actually showed that we can easily leapfrog from the lower part of the Bloom's taxonomy pyramid to the higher ones, so that students don't just remember or have a basic understanding, but get to analyzing, evaluating, creating new knowledge. So this also brings in a new challenge, right? So if we are really creating a classroom or a learning environment where we are moving them from just remembering facts and a basic understanding of concepts, how do we get them to the higher parts of the pyramid? And so we need to change assessments, right? So from a teacher standpoint as well as a student standpoint, how do we assess this thing? So this is just a model that I'm going to propose. It might seem completely random and coming in the middle, but I've been thinking about this for a while. I think we are stuck with the way we teach and we test because we haven't developed new methods. And thanks to the internet technologies, the ability to share or create new ways of testing and assessing student learning, we can actually go from grades to grids, and I'll tell you what that means. So I think all of us are aware of a percentage, right? So somebody got 90%, 98%, somebody got 35% and they fail or whatever, right? So we've seen grades. Oh, I'm an A+ student. Somebody's a D student, or somebody who has E or F. I think some places it's a F grade, or a description, right? An outstanding, excellent, very good. And what do these numbers, letters, or descriptions really tell students, teachers, or employers, right? If we really care about employment. So I think if we really do a little bit of soul searching, these are very inadequate. Now, I'm not saying there's going to be a perfect system. This particular model serves some purpose, but I think it is definitely, it has several lacunae. It really doesn't tell all of us, whether it be students, whether it is teachers or employers, what these things really mean. And sometimes people fall into artificial or completely unrealistic traps. The two extremes being somebody who is an A+ student thinks that I'm not saying it happens all the time, but there's a possibility that they might think, I've reached, this is it. I can't do better than this. I'm already doing the best. Or sadly, and more, I think that should be very concerning, is a student who is supposed to be a fail or a D student, right? There's a perception that they have learned nothing, right? But I'm sure every student who is in a classroom has learned something, right? They probably won't. What is it and how much is it? Is it sufficient? Is a different question, but it is very hard to say that if somebody is engaged in a classroom, they have learned nothing. So therefore, one potential model could be a grid where every course a student can enter and say, all I'm expecting out of this course is a basic understanding, right? Or an intermediate understanding, or an advanced level of exposure to the material, or I want to become an expert. And also there are other dimensions, right? So is it just comprehension skills, which are basic, intermediate, advanced, or expert, analytical skills, or design skills, or communication skills? You can imagine a student who might not be extremely good at, has an advanced understanding, has really good communication skills, right? I'm not saying they are completely, exclusive, but there's a good chance that some students are really good at communicating things lucidly, but they don't have a really advanced level of understanding. On the other hand, you can easily imagine students who are extraordinarily good and they have advanced knowledge, but they lack communication skills. So maybe a student entering a class is not trying to achieve communication skills, but only advanced level of comprehension, analytical or design skills. Now, this is not a fully formed idea, but I think instead of numbers and descriptions and grades, if we had some kind of a grid system that can broadly accepted, so we get to know more about the students' exposure to material, ability to perform some skills, which are actually a lot more useful to everybody. And that will also help us to design courses and material and experiences that will be much more beneficial. So I will end, actually I'm glad I'll be done in the next few minutes. So I will end with a couple of things, and so we'll have enough time for discussion. So these are some action items. I've specifically picked for one particular one, the fourth item in their action items of vision and change, to engage the biology community in the implementation of change. Promote more concept-oriented undergraduate biology courses and help all students learn how to integrate facts into larger conceptual contexts. Ensure that all undergraduates have authentic opportunities to experience the processes, nature, and limits of science. Provide all biology faculty with access to the teaching and learning research referenced throughout this report and this enormous amount of a wealth of knowledge right now in terms of biology education research. And I'm sure some of you are very well aware of, and encourage its application when developing courses. Create active learning environments for all students, even those in first year biology courses. There's a tendency, I think it's well thought out, but I think it's limited. Broadly speaking, people have a perception that we have to give a lot of information and prepare first year students. But in my own experience, I realized that first year students, and in fact some motivated high school students too, can take care of that information part and basic comprehension part. I think if we challenge students enough, and everybody's ability is different, all students' abilities are different, assessing their student ability to begin with and creating personalized learning goals, personalized experiences can take students much farther in their learning journeys, and encourage all biologists to move beyond the depth versus breadth debate. So less really is more. It's a tendency, I think, across universities, across the globe, at least the parts of the globe I have seen, that more is better, right? It turns out, as practicing scientists or in industry or policymakers, we are at a time in history where we can learn when we need it. We definitely need some preparation, we need some skills. So if students and all of us in the real world, that's how we learn. If we gain the right kinds of skills and a decent perception or a good context, I think we are in a position to actually pick up new areas of knowledge, new dimensions of learning. So I think it is not going to be easy, but hopefully, I don't know, I haven't read the new education policy 2020 carefully, but I think the general idea is to create some of these opportunities. And the last thing, I thought this is from a very recent report from the National Academy of Sciences in the US, imagining the future of undergraduate STEM education. It's freely available. You can download the PDF of this book. So one of the tables presented in the book I thought was interesting. So this is innovations in STEM education. These are 10 of them that they've listed. Applying active learning pedagogies, I'm sure we've heard the last 2 years, an infinite number of times people using the word active learning pedagogies. There's an enormous literature, and there's diversity of ideas in active learning pedagogies also. So we pick and choose what is relevant, what is more engaging, and what can be very effective in individual classrooms or with individual students. Implementing competency-based education, I think that's another repeated word in the last 2 years I've heard across different places. Adopting a multidisciplinary integrative approach. This is again, some of these things you'll see that they have been very commonly used in the last 2 years. But I think the idea to remind these things is to go from just chanting them to actually implementing them. Supporting beyond classroom learning experiences, providing flexible, cost-efficient educational paths to continuous learning. I think this point 5 has been reasonably addressed in the new education policy 2020. Enhancing inclusive and entrepreneurship and innovation. I think that's another thing that Make in India and Atal incubation centers, I believe the whole startup thing in colleges also has brought out into discussion. Point number 7, providing advanced support mechanisms for educational research and development. I think I know of islands of work that I know of. I'm hoping. I think my very good friend Arunan is there in the audience, and as that's one thing I keep remembering. Whatever he would say, he would always say, "It is not that we are few, but we are far apart." So there are lots of islands of really creative work happening, like Kyob, I've at least heard of many different efforts. But if we can actually join hands and learn from each other's experience, I think we can definitely create very strong support mechanisms for doing research about our experiments in undergraduate education, and also come up with some best practices that we can all share. I alluded to assessments, and that also brings about this 8th point, developing new credentials. So I think there's an opportunity for us to think about it and come up with new ideas, and at least experiment with it before full-fledged implementation across the system. Number 9 is support connections with K through 12 and peer learning and mentoring. I think one of the things we miss out is there's a huge divide, and I know it's a social and curricular divide. There's very little connection between high school and college. I've been able to do as a small experiment where I clubbed high school students and undergrads. I believe Cube is also doing that, but I'm not quite sure. And the results are extraordinarily uplifting. I'm more than happy to share those things. So when teams of high school students and undergrad students work together, it was a very beautiful learning experience. And the 10th point, enhancing sharing and dissemination of information. I think with platforms like India Bioscience and Current Science, Resonance, I think there is, and of course, all the blog posts and the ease of creating websites, there is, we are in a better position to share, but it would be nice to have some kind of at least hotspots where we can all come to go to and search for information about research and development and education, new practices, new experiences. So it was a little bit of a disconnected thing, but because even till this morning I was thinking, how would I actually combine all of my experiences in the last 15-20 years, and especially the Samyojana experience. So I put some thoughts together. If they are disjointed, I hope we have enough time to have a Q&A, and I'll be more than happy to clarify questions you have or expand on some of the things I just pointed out in this particular presentation. So I will stop here and take your questions. Thank you very much for your patient listening. So Mayur, it's back to you.
About education should not be something that we teach us to our students, but we do along with our students. So this is what Arun sir was saying. So we can take some few questions. So there are many questions on YouTube. So one of the question is, student is asking that when we talk about reading a research paper, firstly, how do we go about finding a paper that has relevance? And the second part of the question is
So Mayur, I don't know if it was my connection or yours, but I did not hear the second part of the question.
Okay, the second part is the language is very complex in the research paper. So how do we apprehend that?
Yeah, so I think it's a process, right? So I'm glad it is truly a different language. So there are methods. I can definitely expound on them, but if some students are really interested, I think it does look challenging in the beginning. And like, let's say it's really like another language, right? So if we're all comfortable talking in English and comprehending it, certainly if I start talking in Telugu, most people will not understand, right? So it's not very different from that, honestly. Or bringing in mathematics. Mathematics is a classic example. It's another language. It does take a little bit of time to master, but it is not impossible. That's all so if somebody is thinking like, oh, how do I go about this? It is a process, and it's something I cannot explain in like 2 lines, right? But there are very well laid out processes, multiple formats, not just one. Coming back to the first question, I think, how do you find a good paper? I think the answer is within us. So you are walking on the street and suddenly you say like, why is this bird looking slightly different from this other bird, right? That's an interesting question to begin with. Or something many kids probably wonder is like, why are my fingers of different length? Why is that there's webbed feet in some animals, but we have very distinct? These are all very, very important questions. And I think engaging in both in classroom and outside, if we really keep the questions alive, what to choose? I think you name the topic, you name the question, and people have been working on it for at least 200 years. In fact, some of the questions, I haven't seen any new question pop up for the most part. It's just that there is more deeper and deeper understanding. So the only way to, I think the first way to get to research articles is to have good questions and say, has anyone worked on this question, right? And right now, my students find papers quicker than me because of Sci-Hub and other places. So if I actually think of a paper and I I think it is behind the paywall, there are a couple of things. Sci-Hub is, students are accessing Sci-Hub and other places. And sometimes the easiest way today, I think today none of us we cannot complain, especially if you are able to access the internet. It's all out there. You name it, if it's PubMed, or in fact, PubMed is probably a little difficult to go through, but there are better ways of finding research articles. The last thing is, in this particular context, I really benefit even today by subscribing or listening to, reading popular science outlets like The Scientist, Genetic Engineering, and Genetic Engineering newsletter. I think many interesting blog posts are there, and Twitter. If people are on Twitter, there are some handles which are like extraordinarily informative. I'm not active on Twitter, but I do follow a couple of people, and go once or twice in a week, or at least a few times in a month, and check out what they're saying. So, and I'm saying this with confidence because some of my students, in fact, the students if I meet them for the first time, even first year students, I throw in a few questions and they actually come up with some interesting, deeper insights. And then I basically tell them to go to some of the, many of the scientists. Let's say you take an article in The Scientist. There's usually a link to that paper they're discussing. So that's one way. If not, I think we are all there. It's just a matter of reaching out and saying, hey, can we discuss? And I think this can literally happen over one cup of chai or samosa. It will actually, many of these things can happen much more easily. And since we are not talking specifically about that, I'm going to stop there. But definitely there are many ways of addressing the first question, how do you find a research article? And the second question, how do we tackle it because it's a new language? Doable, and I have many examples. So if many of these students can do it, all of us can do it.
Yeah, so there is one more question by Arunan sir only. So he is asking that how about promoting context to curriculum in a course structure most often undermining context?
I don't quite understand the depth of this question, but I will attempt to answer this. So if I do bring in a research article, how do I connect it to the curriculum? So I'll give you the example of the same. Is that correct? Mayur, correct me if I'm wrong.
Yes.
Yeah. So the paper I gave to students is what was it? Adenocarcinoma. Let me find out quickly. I forget the title of the thing. The paper was that I gave is pancreatic adenoductal cancer. Right. So these students have been in college for the first time, and it was about 1 month. So when I gave this paper, they all said, "Sir, what is this nonsense?" I mean, they didn't say nonsense, but they all said, like, this doesn't make sense. And one student came and said, because if you look at the paper, there's a lot of microbiome, microbiology, microbial genes and stuff like that. So one student came to me. Remember, this is a first year student. She comes to me and says, "Sir, but I'm a genetics student. Why are you giving us a microbiology paper?" I said, like, you already specialized in the first year itself. Like, you just came to college and you're already a microbiologist more than a geneticist. The funny thing is, there is so much. I wish I actually had student feedback. I didn't put enough of it. But they came back and said, "Sir, now classes are so much more easy for us to follow." This is when a teacher says something. In fact, after the second round of our papers, one student came back and said, "What is this, sir? They talked about CRISPR in one sentence. There's so much more to know. I already know so much more about CRISPR, and why is that they mentioned CRISPR only in one line or one paragraph? For that matter, the whole idea of 16S rRNA profiling or PCR. Like, even if it takes simple ideas like PCR or Western blot or things like that, what the context to curriculum is, some of these research articles provide an amazing context. They create a platform. Otherwise, all of these things that we read in textbooks, they're so disconnected in some sense, right? There is a flow for sure. But let's say, I mean, it's not, I think many of you might agree, and I've done this survey so many times. So I go to students and I ask, "So what do you like about biotechnology?" So Mayur, do you want to take a guess? What is the common thing that I hear?
Maybe the molecular, the techniques and all people learn.
Exactly. So the most recent one, this is from, and I've done this several times, but this is, I remember this clearly. So I said, "What do you like in biology or biotechnology?" And they say, "I like PCR, I like Western blot, and I like flow cytometry," something, right? And I'm like, why? Right? PCR was not always there, right? Western blot was not always there, but there was biology all the time. So I think if the context is there, I mean, basically a research paper comes up with a problem statement, and then they say, how do we address this? These are questions we have, and since we want to address them, we are going to use a set of tools. And they also come up with, especially in the discussion of a good paper, they talk about the limitations of it. So I think there's a good understanding, or at least there's an opportunity for students to say, "Oh, we have a better model, but we don't know all the details," right? And so coming back, very simply said, I think a research article already has many things in a storyline that curriculum can easily plug in. And coming back to the PDAC paper, we actually touched base with simple ideas of microbial diversity. We talked about genes and how gene expression works and therapies. So there are at least I can list about 10 different topics that are there in microbiology and genetics textbooks that are easily connected without difficulty. I can connect them to the curriculum. Now, I have to definitely say that there is slightly, so there might be there might be topics from first year, second year, and third year, right? But I have not seen any student complain after the exercise that why are they telling us things that are in the third year? Because there's nothing called first, second, third year biology, right? It's just biology. And so for the ease of instruction, maybe we divided them, but I think the research paper actually brings back all the unity of biology for that matter. So I don't know if I addressed it, but there was an exercise we were trying to do. We were trying to develop labs where we do something I didn't talk about as something like Cube. I've been doing cures, course-based undergraduate research experiences, not very different from the kinds of experiences that people in Cube have, but we were trying to. That's something I continue to do, where in one semester we do a research, we explore a research topic, right? Actually do experiments in the lab. And I'm trying to do that even here. I'm currently in Delhi and I'm trying to experiment with that, where the idea is not necessarily understand every bit of it, but get an idea of what research is, from an idea to getting results and presenting it, right? Something very simple. So when that happens, and coming back, I don't know. So yeah, we are trying to connect when somebody does, let's say for example, 16S, we published a paper recently and I can share that with you all. So let's say you take microbial 16S rRNA profiling of microbial populations. You can do serial, in terms of techniques, right? We do serial dilutions, gram staining, genomic DNA isolation, PCR, gel electrophoresis, restriction fragment length polymorphisms, looking at sequence bioinformatically and doing going back from sequence to the gel information, right? That's already like 5-6 experiments in the curriculum, but now it has relevance, it has a story. And I don't want to talk about it. If somebody is interested, I can ask the students who are part of this to actually tell their experience.
Yeah, it answers what Aruna sir was asking. I have a different question. If, for example, this process, what you're giving research papers, the first thing which I want to ask is what is the motivation for them to read it? For example, there has to be some motivation. Otherwise, it will be like some sir gave us the paper and we have to do it and we have to present it. So, is it not good to, for example, that I will just share an experience from the queue. So there were these students were there, school students, and they were trapping these fruit flies and doing this experiment. And while doing this, they were asking questions on circadian rhythm and activity pattern, and all seniors were doing this, and they were also involved in the discussion. These were school students from municipal school, 8th, 9th standard students. And in 2018-19, I guess, a group won Nobel Prize in Medicine and Physiology for working on activity circadian rhythm in Drosophila. And when they heard the news, they were shouting and they said, oh, yes, we won the prize. Okay, so while working on this kind of project, they have a motivation to read, let's say, to read the research paper. When you are giving this research paper, what is the motivation in this?
So that's a very good point. I will tell you the honest story, right? So I was talking to the teacher and I said, I think let's do this experiment. And the first motivation was, she said, okay, we have an internal 10 points. I can assign this particular activity to the 10 points in the classroom. And let me tell you honestly, that was a big flop, because the first day 90 students were in the room. But I would say, I didn't know until later on, I realized that I would say, probably not say, I know that, right? Eventually, only 24 of them persisted. The rest of them dropped out. So the 10 points was a useless, because they did not want to be there. Now, in order to, I told the teacher that, let's do it with a very small group. Let's be not exclusive, but let's keep it as a voluntary thing, right? Whoever wants to come. Now, Mayur, really honestly, I don't have an answer, but I'll tell you what I learned from this process. So they were students, and I think the only way is just keep doing this experiment and let's see how effective it is by the results. And the reason why I say this is, so there were 2 or 3 students, they tell me, and I'm more than happy to introduce them to you, they were really not interested. They really came for the 10 points. And they saw something happen different. And there were 2 kinds of students. It was different, and some of them were enamored, and they were like, this is crazy, and they left. Because I told them, look, if you're just going to be bodies in the room, you might as well go do something that is useful to you, or just go home and sleep, right? Doesn't matter. But if you're here, I want you to engage. Some of them got scared. Some of them did see a point. Some of them, they were just curious and they stayed, and they keep telling me if that didn't happen, two things: if the experiment didn't happen, and if they just did not stay there out of curiosity, they would have missed a lot. Now, when I announce Samyojana, I don't even have to work hard. There's already word of mouth that there's a lot of fun, and I'm not saying everybody shows up, because first year students, for example, they have a lot of other concerns. They are dealing with a lot of issues. But I think there's at least about 5 students who go like they are crazy, and they go into college and tell every student, like, if they're not reading a research paper, they are basically missing out a lot of fun. And I think that's a great motivation. So I have now a third generation as in the third iteration of experiments. Some of these students on day one, they are like, I want to be here because I heard so much about it. I really want to experience this. And I think it is like that, right? So we keep doing these experiments, and if it is, I keep telling myself, if it fails, let it die. It's okay. It's not the only experiment. So unless, and I think, the whole growth mindset and other thing, right? I don't know. The only true thing is intrinsic motivation, I feel. Any extrinsic motivation will die at some point. Or it will transform into intrinsic motivation. So I think research articles, discussion about research articles can bring about a lot of intrinsic motivation, because I can show you, student after student, they feel empowered. In fact, one of the students, I cut out that piece in this particular video because it was becoming too long. I had too many nice bites. One student said, presenting this paper made me feel powerful. And another student said, I never thought I could talk in front of people. Right? I never wanted to show my face on the screen. But after this, I really want to show this. I want to showcase what I've learned and understood. And so I think these student stories are one motivation. And as instructors, if we can, the first carrot, if it is a 10-point thing, so be it. And we can at least introduce it and see and make it an interesting experience, right? So it seems like a compulsory thing, but if we can make it a fun activity, why not, right? So, but I haven't found the perfect solution for, what is the perfect motivation, I still don't know.
Yes. We have, so as we are getting close to the time, but we will take one more question, and the rest of the question I can mail you. So this question is, so there are 3 questions with the same gist. So they're saying that what is reliable source, how to distinguish between reliable source from the fake source.
So I think Deepa Chirani is also, she's raised her hand quite a few times. So I took that question, but Deepa, do you have another question you can ask?
So it was truly an interesting and a thought-provoking session, sir. In fact, it's truly a need for, as sir you mentioned, it's a need for concept-based programs, and also to create an active learning environment for the students. Sir, I have one thing to ask. Sir, you introduced about the book festival at your end. So can you just highlight or tell us something more about the book festival that you all have over there, sir?
So there are amazing books out there. Fortunately, some books about Indian stories are coming out. Sadly, most of these books are, for example, probably many of you have read or heard of, is Jim Watson's Double Helix, right? If you've read it, if you haven't read it, I strongly recommend you read it. So we've done this with Garware College in Pune as well. It was a very interesting experience. I haven't found anybody who did not like that experience. So I'll list out a couple of books in the next 30 seconds. Just for the DNA double helix, how did they discover what happened? Watson's double helix, Crick's What Matt Pursued, Wilkins's The Third Man of DNA, Brenda Maddox's The Dark Lady of DNA about Rosalind Franklin, and then you have, that's 4 books I think out there, right? And you talk about biochemistry, Arthur Kornberg's my love for enzymes. I have a whole list, right? I mean, I can send you that. And fortunately, I probably cannot say it out loud, but many of these things are available online. No, I mean, ideally, we should buy these books for sure. Hopefully, we will be able to access them physically. A Feeling for the Organism, I see about Barbara McClintock. There are amazing biographies and autobiographies out there. So what we do is each student, they pick, I mean, there's a process we come about so that people are not fighting. But everybody picks a book. They read it for a semester or 2 to 3 months. And we all come together and everybody gives, depending on the number of students, it's a 5 to 10 minute review of the book. What did I like about the book? Why should anybody read it? If I didn't read this book, what would I have missed? And I'm more than happy to share some videos. It is emotional. Some students were emotional about this, right? They were like, I felt about this, they became part of the protagonist of the book. They saw science as a social process, it's somebody's life, right? So there's some really good stories. So there are many ways we can do it. It's just that I think I don't even have to. There were students who were scared of reading books, but they picked up. Some of them decided at the end that they didn't read the book, or they were scared of presenting it. And then finally, when they heard other students present, they said, "Sir, we missed an opportunity. Now, can we do it again?" Right? I think that emotional connect with the process of science, I think, was the reason behind its success. Coming back to, Dr. Deepa, I will actually talk to you. If you're interested, we'll talk more about it. I know it's only 2 more minutes. To answer the first question, what's a reliable source and what's a fake source? Peer-reviewed journals is a reasonable good source. I'm not saying that peer-reviewed journals don't have fake data. I'm not saying that, because that is a problem too. But so I'm not a big fan of cell press necessarily, right? But it has a reputation. Let's say Cell's, Nature, and even though there are several papers which are retracted every year with fraudulent data, more or less there are big stories. At least the community accepted them as good stories, right? So peer-reviewed articles from established journals is a good place to start. If they do find fraudulent stuff, that's another great exercise. Even in these great journals, how are these things happening, right? It's, after all, part of the scientific process. I mean, at least modern social aspect of the scientific enterprise. So I wouldn't be too worried about the fake versus this, but it's always about, I talk about 2 things, assertion and evidence, right? Or an inference. If a scientist is making a claim, what is the evidence and what is the authenticity of the evidence? That is all we ask, right, when we read papers. So that is a practice that I expect students to follow all the time. I know it's 12 p.m., so I will stop here, and it was a delight. Thank you for this opportunity, and thanks a lot, Mayur, for reconnecting.
Yes, it was amazing lecture, sir. So rest of the questions and comments, I will mail it to you. And over to you, Saida.
It was really interesting talk, sir. We have, of course, we'll take cues from it. As of right now, I'll hand over to Shrujal to give vote of thanks.
I would like to express my heartfelt gratitude to Dr. Anil Challa sir for this commendable talk. It was such an honor to have your gracious presence with us this webinar. It was really great to hear about the Vision and the Change program, which was based on the basic concept of scientific methodology. I personally found your vision, that is to replicate in this India with a conceptualized 6-week program. That was really very motivating. Thank you again for taking time out of your busy schedule and enlightening the crowd with your educational talk, which will definitely help us on our journey ahead. Over to you, Saida.
Thank you, Shrujal, and thank you, Dr. Amruta Naik and Professor Anil Kumar Challa, for enlightening us with this amazing talk. With this, we have come to an end of the seminar for day 1. But wait, picture abhi baki hai. We will be right back after a short break with exciting events like Speakers Forum, wherein participants has to speak on a given topic for about 1 to 2 minutes.
After which, we will also be looking at some of the social media based events like Reloconic and Memers Mania. So grab your popcorns and we'll be right back after the break. And I would request everyone who's present in the meeting to please turn on your cameras. Let's capture this moment. Sachin, can you take the screenshot?
Yes, I'm doing that.
Anil sir, we want a photograph. I guess is yeah.
Here's the.
Are we good? Done?
Yeah.
Yes, yes. So Saida, we have a break now.
Yeah.
Can you
Yes,
announce
sir.
that? Yeah. Till what time we will be back?
We'll be back by 1. So see you after
It's
the break, everyone.
Yeah.
We'll be restarting the event at 1.
Yeah. The same link.
So welcome back. As we have gained valuable knowledge, now is the time to get a little more excitement with our exciting events. So I will like to call Saket to start our event, the Speakers Forum. Over to you, Saket.
Thank you, Ria. Welcome you all. Good afternoon, everyone. We will be starting with our event Speakers Forum. But before starting with our event, I would like to introduce our judge for today's event, Dr. Savita Patil, Associate Professor and Head of English Department at Elphinstone College. We welcome you, ma'am. Now, to make our participants well aware with the event, I would once again like to repeat the instructions. The preparation time of 5 minutes will be given to each participant. The speech should be delivered within 1 to 2 minutes. Stay relevant to the given topic. The judges will evaluate the participants on the basis of speaking fluency, staying relevant to the topics and content. Now, calling out our first participant, Meghana Savan. And her topic would be effects of lockdown on the environment. She'll be joining in the meet within 3 minutes.
Participant is there?
Yes, sir, she's preparing for her speech.
Okay,
Yes,
fine.
I'm here, but I got my topic, but I have 5 minutes to prepare for it, right?
Yes, 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 yes.
Yeah, sure. Thank you.
starting in 1 minute, and all the participants should be turning on their cameras when they are speaking. Thank you.
Hello, should I start?
Yes, you can start. Turn on your camera and.
Okay, okay. Am I visible and also am I audible? Please let me know if I'm not, because
Yes, you're
the
audible.
network might fluctuate. Yes. So, good morning, everyone. Today, my topic is effects of lockdown on the environment. So, past 2 years has been very difficult for us, but the positive impact of it has been on the environment, on the nature itself. There were many news articles which showed that how the lockdown, every all the human population being locked in their houses has impacted the nature so much. For example, olive ridley turtles and the percentage of them present were increased. The air quality index, which has been low in Delhi since like forever, was getting better. So according to me, this has proved that we humans are responsible for what is being going on with the nature. Be it the presence of microplastics in deep down in the oceans, or the air quality index being down. This is what maybe the nature is trying to tell us, that the human beings are responsible for whatever bad is happening with the environment. And I would like to say that this is just an example of what changes we should include in our daily lifestyle. Humans being locked down in their houses has impacted the nature. Like, the rivers are getting clearer, less pollution in the rivers, the air quality has increased. Wild animals, the fragmentation which has been happened because of industrialization and urbanization, they are feeling free.
Megan, are you there?
I guess there's some network issue at her end. We'll wait for a minute. If not, then we'll continue with another participant. Is it okay, Sakit?
Actually, our timing is over. So we'll be moving with second participant, that is Mercy Fernandez. Her topic would be, how does technology affect our sensory organs? I would like to ask Mercy to turn on her camera and start.
Just a second, please. Am I visible and clearly audible?
Yes, you're audible.
Okay, so the topic for my today's speech is, how does technology affect our sensory organs? Basically, what is sensory organ? We have five organs: eyes, ears, nose, tongue, and skin, which are an integral part of our life, as it helps us to provide information that are basically specialized organs which help in our living. They provide required data for interpretations throughout various organs, and technology nowadays is advancing day by day. As we know, technology have both its effects that are advantages and the side effects. So the effect of this technology in our eyes, basically now it is a period of the lockdown where we are just going through online lectures and all. We can see the mental status of that student, like from the whole day to night, one student is present in the screen. Even books are now available online only. So in some way it helped because the cost of things have been decreased which we needed, but the eyes, nose, or ears, they are like, we do not get time to just recover from the stage. Like day to night we have all the same routine with that screen. Like we can see now also we are in the screen, stuck between screen and having this event. So I would like to conclude that technology has basically helped us only, but it also had the harmful effect to our. It also had an harmful effect to our living lives and to the sensory organs, which help us to be a part of this world in a more beautiful way. Thank you.
May I interrupt for a minute?
Yes, ma'am.
Hello. Yeah.
Yes, ma'am. Yes, ma'am.
I request the participants. Request them to start their cameras when they speak. Otherwise, it will be difficult for me to evaluate. Let me see whether they are extemporary speaking or what kind of it is.
Ma'am, my camera
Okay, so
is
you have to
on.
keep your camera on. Yeah, till end. Am I right, organizers?
Yes, yes, ma'am. Yes, ma'am.
Yes.
Sachin, can you remove the banner so that ma'am will able to see their faces?
Yes, ma'am. Actually, the current participant is having the camera on. Maybe because of screen share, we cannot see her, but her camera is currently on. Yeah. Throughout the speech, her camera was on. Even for the previous participant, it was also on. Can you see her now?
Okay, beta. Thank you.
Yes.
Yeah.
I will request
Consider-
Sachin. So whenever speaker is speaking, please pin them so that ma'am will able to see them. Okay.
Thank you, sir. Thank you. Thank you, Mercy. Fernandez. Now we'll be moving to our third participant. Actually, our third participant is not present in the meet, so I would call our fourth participant, that is Aditya Joshi, to start his topic with the social media making us antisocial. Thank you.
Good afternoon. Am I audible as well as am I visible?
Yes.
Okay, thank you. So, good afternoon. As the topic name suggests, that there are two contradictory statements, or we can say words in the title. We can say, is the social media making a person or making us antisocial? So, social media and antisocial are these two contradicting terms, we can say. So first of all, I would like to say that there is no complete white or complete black in any of the population. So, but what we can say is that there is a, we can see, increasing rise in the tendency of becoming antisocial while still being very much active on the social media. Like for example, we can see in day-to-day life, if there is a family function, all the people will gather and they will take the selfies and they will post. But are they taking the selfies just for posting, or they are really they really need to cherish that particular moment that they are capturing it? So well, even in friends or in the relatives, or even in the, if someone is going to the picnic or outing in the beautiful nature, seeing beautiful sceneries and flowers, they will just take the picture and post on the some social media site and gaining their likes. So it is like the people are more concerned about the status on the social media, but not the skills on the actual social level. Of course, as I earlier mentioned, we can't say all the population is the same, but yes, the increasing trend is surely seen. So majority of the social communication skills are very much good on the social media platforms, but actually meeting a person, getting known their thoughts, spending well time, this kind of trend is just decreasing. Like in few years back, the friends would meet and just go for a short walk in the evening, but now we can say they are happy just by being in touch on the social media platforms on online modes. So yes, there is the rise. We can completely see that. So I would say the social media is making us antisocial, not because it is making us, but rather we are getting or taking that thing in that particular way. We can balance both of them very much surely. Thank you.
Uh, Thank you, Aditya Joshi. The next participant is Archie Paneri of the topic depletion of ozone layer.
Okay, am I audible? Hello?
Yes, we can hear you.
Okay. Okay, so my topic is depletion of ozone layer. So ozone layer depletion causes increased UV radiation level at Earth's surface, which is damaging of human health. Negative effects include increases in certain types of skin cancers, eye cataracts, and human deficiency disorders. So indeed, ozone layer is protecting our Earth from UV rays, just like and UV rays is basic. Okay, so I'm going to give it in a metaphorical way. UV rays is basically a negative ideas and thoughts which we face from our day-to-day life. So, uh, as an example, I just like to quote it in a phrase. It's like, stop worrying about other people understanding you. Get in touch with yourself instead. Focus on what makes you happy, what makes your soul feel at peace. You're your biggest commitment. So start loving your flaws, your awkwardness, your weirdness, your intensity, your vulnerability, your everything. Life becomes so much more fulfilling when you're simply yourself. The world keeps shining whether people understand you or not. Give it a big smile to that. So instead of going it in a scientific way, I did it in metaphorical way. Thank you.
Thank you, Archie. Now, our next participant, Joey Gupta, with the topic, do the benefits of vaccination outweigh the risks?
Am I visible?
Yes, you are visible.
Am I visible now?
Yes, yes.
So should I start?
Yes, go on.
Yeah. So as every coin has its two sides, and also we know that the head is always overweighing the tail, the decision should be taken by inform the balance between the potential benefits and the cost risk. Or you may have probably heard about the Miami doctor who died after getting the Pfizer vaccine. So what does these vaccines actually do? These vaccines, such as specifically mRNA vaccines, such as Pfizer vaccines and Johnson and Johnson vaccines, these vaccines are specifically mRNA vaccines, which have a tiny piece of mRNA which stimulates the antigen and further forms the antibody, which helps in boosting the immunological reaction. After these boosting, so do you find something very peculiar? That is the introduction of a new antibody. So this in turn rises the temperature of the body. But does that actually mean that the vaccination is wrong? No, not as actually. Because vaccination provides antibody which helps in finding the virus, for finding and also helps in fighting to the virus. How does it help? These antibodies react with the virus or the foreign pathogen and burst them and don't allow them to cause any sort of adverse effect in the body. Now, as it was also found out that in military, some professionals like they got a very adverse effect, but these they didn't die. So it doesn't mean that if the coin has something, if the thing has some adverse effect, it should not be taken. So, as the COVID, I would like to conclude it in a way like it need not to be Thank you.
Thank you, Jui Gupta. Now, our next participant, that is Khan Amir Nisha Ayub, with a topic Advancement of Science in Recent Years. Thank you.
I'm audible.
Yes.
Good afternoon to all. My topic name is Advancement of Science in Recent Years. Science and technology are interrelated. As science progresses, technology advances. It is a growth in technology is an indicator of development of science. Advanced technology has made our life more comfortable and restless and more lasting. Uh, we use some refrigerator, air conditioner, mobile computers, and television. They are mm, they are boons of technology. In a rocket science, imagine a rocket reaching up to Mars and the level of modern technology and sciences work on it. As humans have gradually developed science like mechanical, medical, botanical fields, etc. It becomes a key uh, for teaching technology of advance. But one thing has to be remembered: science and technology technology do not hazardous to us, and it is used in in wars like bomb explosions, modern artillery, and so as long it is a good purpose for use. Now the technology is developed more probably. During this pandemic situation, we use different types of technology for connecting to other people. We gain some skills, and we have we gain some skills during this pandemic situation. Uh, we uh, hmm, we encourage medicine for vaccination.
Thank you, Khan Amir Nisha Ayub. Moving with our next participant, that is Raghav Thakral, with his topic Ethical Issues Concerning the Use of Laboratory Animals.
Yeah, well, am I audible and visible?
Yes, you are.
Okay, well, thank you. Well, good afternoon, everyone. My topic for today is ethical issues concerning the use of laboratory animals. Well, what do we mean by ethics, first of all? Ethics means moral goods and rights, which we know that. Ethics basically concern that as a human being, we all have feelings, and we all feel pain, we feel pleasure and all. So in the same way, the ethical issues regarding animals concern that do animal feel the same kind of pain and pleasure. So yes, definitely they feel the same kind of pain and pleasure as we do. Tomorrow, last day tomorrow, I saw an animal being run over by a car. So and he was like in a very bad state. But now here the issue is regarding the use of concerning the use of laboratory animals. So laboratory, there are some ethical issues, like do they have the same kind of rights as we have? Basically the researchers are responsible there to access the to access the affected effect of the laboratory animals. What kind of effect they're gonna have on these animals? Researchers are also responsible for ensuring the use of laboratory animals to not endanger the biological diversity. And it is very important that they see how diversity is actually being affected. these at some cases we have heard of that these laboratory professionals they actually endanger these animals life, and after that they get the results. In order to achieve betterment for the humanity, they kill those animals. They treat animals in a very bad manner, in a very inhumane manner, and these are some very prominent and very basic issues that concern or the ethical issues which concern the use of animals in the laboratories. And at last, I would conclude that these issues must be seen uh, in a very important manner, in a very important way.
Thank you, Raghav Takral. Now, moving with our next participant, that is Kirti Prajapati, with the topic Artificial Intelligence, Boon or Bane.
Good afternoon, respected judges and dignitaries. My topic for the day is artificial intelligence, a boon or a bane. So, as we all know that artificial intelligence means the science of making machines that are modeled after human intelligence. These machines are programmed to think and work just like human beings. Artificial intelligence is not just about the robots, but it has a broad meaning which includes perception, learning, reasoning, and everything. Artificial intelligence is just like a model of human knowledge in the devices which work on the instructions of humans. Not only this, but they can also mimic the actions of humans, including learning and problem solving. Artificial intelligence can be applied anywhere. This technology is involved in healthcare industry for treating patients. It is also used in games like chess and ludo, where you play against the computer. That is also artificial intelligence only. So today, artificial intelligence motivates the innovation of new machines and gadgets which can make a task much easier. Also, it helps to do the work effectively and efficiently. The work of a machine is much more accurate than humans. So it is a blessing to us, and it is also a boon kind of to us because it eliminates the necessity of humans to perform a task. As we know, artificial intelligence is a boon to humanity as it eliminates the need for humans to repeat and perform a task with perfection. So the machine can perform all the tasks which a human can do. It can give a proper result with expertise. It provides time for humans to work on something more creative and useful. Also, it is a perfect combination of creativity and technology. And it can also help us in smart weather forecasting. And it is a blessing to us because, as we know, it can give accurate results. And I would like to end it with a quote that predicting the future is not magic, it's artificial intelligence. And artificial intelligence is a tool, not a threat. As we use it, we can get it the way we want. Thank you so much.
Thank you, Kirti Prajapati. Moving to our last participant, that is Asta Gajanan, with the topic Ill Effects of Human Activities Accelerating Species Extinction. Thank you.
Anthony William once said that humans have the power to save animals or make them extinct. It's in our power to decide. It's our choice. Do you know there are 99% of animals who have gone extinct? According to the biofossils record, we have known that six mass have occurred, which is due to human activities which cause a large extinction. The main cause of extinction is human introduced exhausted species, which breaks the web cycle of the animal, which also cause a large part of extinction. Brown tree snake is an example of this extinction. Over harvesting of fish, tree mining also called a large part of extinction. And pollution, which is of chemical, air, noise, or many things, it is also part of the extinction. Do you know global climate change is main part of this extinction, which is also due to burning of fossils. And last but not the least, we have many extinction species such as red panda, brown tree snakes, tigers, non-susceptible species, and many more. We humans have caused a huge hazard to the extinction species. So please use your power to make and to save the animals, and do not cause a rate of extinction to them. We are now trying to save many animals and stop the problem of extinction. So let's take a step into it and do not disturb our plan. So with this note, I would like to end my topic.
Thank you, Asta Gajanan. And done with the all participants now. I would like to extend my hearty gratitude to our judge, Dr. Savita Patil, for giving us a valuable time and helping us in deciding our winners. Do join us on 2nd March for the result declaration. Thank you. Now, I would like to hand over the mic to Saida.
Thank you, Saket. Well done, all the participants. It was great to hear you all. Thank you. Thanks to our judges for joining us.
Yes. So, can we have some words from our madam? Savita ma'am is around?
No, sir. She left.
Okay, she left. Okay, fine. Yeah, so you can continue.
Later.
Yeah. Yes, Saida.
So now let's take a look at some of the memes and reels made by our participants. Sachin,
It's going too fast. Can we slow it down?
Yeah, can we have a look at one of each, because it's very interesting. Can we read it out here? Okay, you make my heart skip a beat. That's really funny. Can you go on to the next one? And when I got a random compound during semi-microanalysis, I was not expecting this. Okay, can we read out the next part? I cannot read it because of the screen shared. Sachin. Yeah, can you get full screen? The fish decided. Okay, the fish decided to walk out of the water one day, and now I have to make memes. Okay, that is really funny. Yeah, we are making them make memes out of it. That's nice. This is how I keep knowing. Even
algae is a relationship with fungi.
Okay. This is making science fun.
What is bittern? Adeline. Oh.
Oh God. Adenine thymine. When the world realized that the body needs glucose to function.
So they started lay food. This.
It's really nice.
मैं क्या करूँ फिर job छोड़ दूँ.
This was very popular back then. मैं क्या करूँ job छोड़ दूँ, and people are doing great job putting into memes.
The normal virus, the mutated version of virus, yeah.
So the lockdown never ends because of this.
Yeah.
Can your Bhabha went this time?
This reaction
happens with us.
Yeah.
We can relate.
Totally. The cell organisms will mitochondria, yeah.
Powerhouse of the cell. 7th standard biology. That's nice.
When you're accidentally break the cover slip while performing the experiment?
Oh, while using the microscope. Oh, I don't know how well the online people can relate to this, but yeah, I'm definitely relating to this. When we ever use the microscope, we're not able to exist and end up breaking the cover slip. Oh, it's trauma during the practical exam.
What is this?
I used 10 more tonight. This happened, you know, I think in my Viva, in first year, it was Namrata ma'am who asked me what is funny.
Yeah, that question was to me too.
High five on that. It's definitely relatable. I know the process, but I don't know the process name.
Yeah.
Glycolysis, I know. Okay, next.
What if the fell falls?
Gravity. Oh, nice. Really nice. I thought it's in, you know, this is like making science entertainment.
Yeah.
If you study like this, honestly, back then, it would have been even like a better way of studying. You'll remember this meme.
Yes, you will also get marks like memes, nothing.
Yeah. Now, it's good that we are having events now. You'll get marks for memes as well. You'll get awarded for it. That's nice. Can we look at reels also, Sachin? I must say the participants are really talented to pull it off.
Is the screen visible?
Yeah, the video is still loading, so we'll wait. Okay, the screen share stopped. Sachin, can you just look over?
Yeah.
Again, it stopped. I guess there is some technical issue. Sorry for the delay.
So what's happening? Are we seeing the reels?
No, sir.
I guess okay, yeah. Now we can see this.
यादों के पुराने एल्बम में छुपा के रखे हैं हमने वो दिन फलक में बड़ी के बनी सी बचा के रखे हैं हमने वो दिन ना किसी मंजिल की फिक्र थी जिंदगी जीने की उम्र थी और दोस्तों से उधार के तुझे वो जिंदगी का दिन थे वो जिंदगी का दिन थे Sacred Sunday. Here she go, either a little too high.
Oh wow. That was really awesome, and I must say the participants are really talented to pull it off, the memes as well as the reels, for sure. Like, and there were some moments in there where we could relate
Yeah,
working in
actually.
the laboratories, which a lot of people are missing these days. And thanks now, we are starting offline again, so back on track.
Okay, so this were really awesome, well done participants.
The reels were
And
very amazing, I guess. They were very good.
Yeah,
So we should applaud for their creativity, and of course, memes were also very interesting. Yeah,
definitely.
I'm sure people here, and you should have spread this, which is on the Primer's Instagram, right? So
Yes.
that, yeah. Yes, over to you.
okay,
Yeah,
so
it was a really nice session. I mean, it was fun filled, memes, reels, both very entertaining and good. Even the extempore speech had a scientific temper in it. So we really had a great day today with lectures, with such enlightening lectures in the morning. Well done, well done, Primus team.
Thank you, ma'am.
thank you, ma'am. So participants, don't forget we have bought your 1 day bonus to get as many likes as possible, as the count of the likes will stop by the end of 1st March.
Now, as we have reached the end of day one, it is surely not the end. We will be back again with yet another talk by Dr. Subodhit Sen and some more exciting events. So, in case if you have missed out and would like to register for the talk and the event, please follow the link in the WhatsApp and Telegram group, and the same will be available in YouTube description box.
So on this note, we end today's session. See you all on the day 2 of the primers, that is the 2nd of March at the same time. Till then, have a good time. Thank you.